Hey everybody, good morning, good afternoon, good evening from wherever you may be watching. Welcome back to Schmidt Ocean Institute's Divestream. You are watching a live camera feed from ROV Sebastian. We just launched, say about 30 minutes ago. It's a beautiful day out here on the high seas, and we are here at Station 3 of our expedition. Our new expedition is called Animals as Living Bioreactors, and if you want to know more, I will be posting a link to our website where you can read up about what kinds of sampling we're doing. So it'll be in the description shortly, and it will also be a Nightbot command. Here in the control room today, I have Doug and Leo piloting the ROV. We've got lead ROV supervisor Doug as pilot, and then Leo as co-pilot on the manips and camera controls. And my name is Sophia. I'm the multimedia technician. And behind me, I've also got Ian, who is our marine technician, helping to make sure that all the sensors are running and all the navigation and all the other equipment is working well. It's our first dive, so sometimes we have little hiccups here and there. Yep, we have some people in the chat. Good morning. They're asking where we are. If you type in the chat donde or location with an exclamation mark, you'll be able to get a live link straight into the chat for where we are. And as always, we are monitoring the comments on YouTube and on Twitch, so feel free to post your questions there. We have an entire control room full of scientists that are really happy to answer them. And I think up in a little bit I'll put up the map of where we are. But right now we're aiming to do some midwater transects. So the ROV actually won't be hitting bottom today. We just recently were adjusting some camera controls in order to start conducting a video transect to see if we can find any animals.
But like a discarded house? No?
Usually they have more gunk on them.
See a little soul misses flying by.
Or the ones I'm used to seeing.
Good morning, good morning. I see we also have some viewers on Twitch. Thank you all for joining us. Murphy, you want to say hello? All right. And with that said, we have quite the interesting dive ahead of us, and I have a wonderful watch lead up here up front with me today. Her name is Colleen Durkin, but of course I'll let her introduce herself in just a minute. And I'd like to extend a warm welcome to everyone and thank you all for joining us.
With their eggs, so did the eggs in there? Yeah. So cool. It's bad if you're sorry.
All right, Colleen.
Mm-hmm.
I've unmuted your mic, so feel free
Okay. My name's Colleen Durkin, and I'm a oceanographer from the Monterey Bay Aquarium Research Institute in Moss Landing, California. And right now we're doing a quantitative transect at 127 meters. Somewhere around 125 meters, we're just slowly flying the ROV forward to get a quantitative survey of the animals in the water at this depth. So we're about, let's see, we have 7 minutes left out of the 15 at this depth. And then we're gonna go deeper to survey at increasing depth intervals. And the point of this is to complement the Mach Ness net tows that a lot of people on the ship have been doing, a different way of observing animal populations with depth. So the ROV and the nets get really different views of the types of animals in the water. There's not that many animals at this depth, actually. A lot of marine snow. But I have to say, I'm used to Monterey Bay, which is like crazy productive. So maybe there are a lot of animals here. I'm just used to seeing like, you know, super, super productive coastal area.
It's also daytime.
Yeah. Right. Maybe as we get deeper, we'll see more. But there's a lot of really beautiful marine snow, which is my favorite thing.
That flashing is caused by one of the lights. We can turn that off.
The light?
Yeah.
Yeah.
It's a little backwards, yeah.
Well, I don't know, because now it's now the lighting's not. Yeah. So is that light just not going to work?
They'll do that from time to time.
Yeah, it would be better to try to keep it consistent, I think. Seems better now, right?
Yeah, it does that sometimes. It's
Okay.
on its way out. Unfortunately, they don't make them anymore, so I don't have a spare for it.
Well, maybe we can, just what you did there seemed to
Okay.
work, yeah.
No, it's good. Look at. All you're gonna do is pay more out towards the.
Well, there's something.
Will we be stopping at this waypoint?
We're probably gonna go a little bit further, Tony. We're doing this all time based. We don't have any waypoints to go off of down here, so
Okay, mate. I'll just extend it, and then if you guys want me to stop, we can do it.
Yeah, we'll move the waypoint a bit, and then yeah, we'll do that. We'll let you know when we want to stop. Thank you.
Okay, mate.
Good.
And this is in a minute and a half. We have a minute and a half left of
Okay.
this.
No problem. They're pretty flexible up there.
Okay.
And then you want to stop and then come down to the next level and
Yeah.
then continue on.
Yeah,
Okay.
but maybe we could, yeah, why don't we do that?
Alright.
And then before we start the next one, let's double check the focal, like try to find something and see
Okay.
what the focal distance is.
Alright.
Or just try to be at the midpoint. It looks, I think it's pretty good.
It
but-
usually is if
Yeah.
you turn the auto off. 'Cause
Yeah.
Otherwise, you just see it hunting back and forth,
Uh-huh.
and I can tell when things get close, they get blurry.
Yeah, exactly.
And then when they're
Yeah, look
far
at
out,
that.
they're pretty blurry as well. And they're
Mhm.
it kind of seems like
It seems like middle.
it does to me.
Yeah, I think
But
so.
I don't have like a slider bar that I can look at that tells me it's the middle. If
Well,
if you
yeah.
understand what I mean.
Yes, I do.
It would be helpful if I did, but
Yeah.
I haven't found that yet.
Mm-hmm. Oh, there we go.
Oh,
Okay.
that's it.
All right, so that's
Tony, if
15
you can
minutes.
Slow to a stop here, please.
Copy that. Copy.
Cheers. All right,
Okay.
go ahead and
So then,
Station
oh,
over
sorry.
here.
And
you want me to go to what?
so the
depth
next depth is 250.
All right, coming down.
Oh, it's like right in my view. Is there a way that it can be like to the side or?
It was fine.
Okay. It's like, do I just have to stare at a microphone or?
Sorry, that's all the picture you paid for.
Yeah.
Sorry.
Microphone.
Okay.
You can go a little faster.
No, but these depths are set.
Yeah, I know.
Oh, cool.
So that's like around.
What depth is that? I can't read. I'm sorry, I can't read.
Yeah, this is 250,
250.
and then this is 500.
Nice.
So we're somewhere.
All right. Well, hopefully we start seeing more animals. 250. That's our next step.
Yeah, and below that we should start seeing more.
Okay.
I mean, this isn't the real time, but this is where we're like that would be the migration, right?
It was changing the
So,
yeah.
okay, Anitra just showed us the EK80 images, which are like acoustic signals of the deep scattering layer, and it looks like there's a big signal of animals migrating down to 250 and 500. So hopefully we'll start seeing more animals as we get to those layers. But I'm enjoying the rain snow.
Also, Colleen,
Uh-huh.
if you watch this picture, that's also just facing forward. It's just slightly down.
Oh,
As
so
we're
what's
going.
this one? That's the one,
upper.
that's the upper
Yeah, that's
camera?
the upper camera, yeah.
Oh.
And I have it just slightly
Sweet.
tilted down.
Yeah.
So just past the jars. So you have
That's
both those
beautiful.
views right within your
Yeah.
within your reach.
It's so awesome that you have these two 4K cameras.
Yeah.
It's amazing. So this one's just going straight forward?
That one's straight forward and
Yeah.
the other one's slightly down.
Okay.
And however you need them adjusted, just say so, because I'm really just
You mean the
piloting by time
direction?
and my
Or
nav screen.
Okay.
Yeah, this one can only go up or down.
Yeah. Okay. And this one is just going direct forward.
It
The
is,
camera?
yeah. You see how that's the angle
Oh,
of
I
it
see.
right there?
Okay.
See how it's kind of straight out? It's just straight out
Is
over
it-
the jars.
Is it coming to the like starboards? Is it kind of angled to this? Oh wait. Oh no. I see. I was looking at the
That's-
wrong thing.
Yeah.
Yeah, I see the camera. Yeah. I'd say let's do that and until Anella tells us to do something
Okay.
different.
Andy, right now we're just going down to 250 meters, and I've been using the auto depth.
Okay.
And then we're just continuing this direction for 15 minutes.
Okay.
And we're just doing video
Tracking it for the
transects.
time?
Yeah, she's keeping track of the time with her timer.
Okay.
And then whenever we get going, we're just moving Tony. So I'll get down to 250, stop, and then I'll give it
Are you
to you.
gonna need to do some mic balance change?
She might want to just see. They're trying to be like mid focal range. I have it in manual right now. It seems to be mid, but not positive.
Maybe
So
when
you
we
might
get
have to
when
mess
we
with it.
when we get to 250, we could try to find something and kind of get a sense of the focus and see if we want to adjust it.
The jar camera has a ground fault, so we just have it off for right now. But that seems to be the only thing giving us fits right now. So pretty good. Actually, if you want, I'll give it to you. There's a bit of current, so. There you go. You're lined up. Cheers.
Yep.
Oh, do you want me to come back after the meeting, or are you good for 2 hours?
No, that's fine. It's fine. Unless it work for you?
Yeah, it's good for me.
Yeah.
All right.
What's your name?
I'm Andy.
Okay, I'm Colleen.
Oh, nice to meet you.
Or maybe we started at 7:45 right now.
So we're trying to dive down to 250 and then start the transect.
Okay.
Is the current different at this depth? Is it still about half a knot? Okay. So what we were doing before was just driving straight into the current at something like 0.2
Okay.
0.2 meters per second. And maybe we could try to like let's just double check that we got the focal depth at the place we want it. So just maybe try going a far. I don't know how you adjust it. So let's see. I guess it's hard when everything's moving.
Much.
Yeah. I think it was pretty good where it was at. So I maybe if we just leave it, it's fine.
Sounds good.
Hey Anella, what do you think of this lighting?
It looks pretty good to me.
Yeah, it
Yeah.
looks good, right?
It's very well distributed and even.
And then what about the iris?
Have you guys been zooming out
is zoomed all the way
Looks
out.
like this is zoomed
No, this is zoomed all the way out.
So I think we did our best with the focal depth, or it's really hard to judge,
Yeah.
but it kind of looks like things get out of focus as they get closer, so
in. I think it's pretty good.
yeah, okay. Stamp of approval from Anella.
Alrighty.
What do you think, Anella? Should we start this transect? Do you have any other adjustments you want to make?
What's the tilt with the camera?
Oh, right now it's straightforward. What do you want it at?
I think worst case is like 20 degrees or something like that.
I think that the constraint here is probably trying not to get the jelly jars in the image, but we could try.
Oh, right.
If we tilt down, what happens? Oh yeah, we're right. They're right outside of the. I think that's about
I
as.
mean, I would just, I guess, keep it what you've had it at for the past 2 months.
That
We only
camera
have done
there
one.
is
We've only
looking-
done one.
That one is looking down a little bit.
I see.
This is going forward.
That I think that's what. I don't think we have any other option.
That's
Okay. Unless you want to look up. No. All right. Then I think we want to just drive straight forward at trying to maintain 250 as much as possible and trying to just maintain the.
That's the auto, so that's what you're seeing right there.
Okay.
Lips.
So then trying to maintain the 0.2 meters per second about, or it was kind of going between 0.2 and 0.3.
Yeah.
So if you're ready, then I'll set
Sure.
the timer. All right, it started.
Great, you ready?
Go ahead.
Hey mate, we're going to move, resume the move again, please.
Resuming the move.
Where's the? Let's see. Lots of still a lot of particles. They're not quite as fluffy as shallower. Siphonophore. Look at all these bigger than 200 micron particles. It's really fluffy marine snow. It's really cool. Mm.
Got your coffee. Bye.
Goodbye.
I'll be back. I've still got another hour.
We're going in layers. Yeah, to 15.
Like an onion.
Our job is just to keep the vessel. And the lower cats,
Oh, some kind of a fish or something.
and switch it off and on again and steady. Some steps. And the T4 is giving up a couple.
Yeah.
And you didn't want water from ECW bottles or Niskin bottles?
No.
Sweet. Something we want to fire them.
And it
What depth?
don't change any.
No
Some
chemicals.
for the tree.
So already set.
Which here's the list of depths we're going to.
Just keep going, don't change it on cameras, don't change it on
The lower list.
Just,
Oh, there's another salp. Is that? Just taking notes on that.
Define spectacular.
Do we want to fire the Niskins during the survey or on the
Let's do it. If you want it at that desk, after we stop, we'll do it. Oh, beautiful. Salp.
Huh? You did one. That's one. Oh, you did this.
Another salp, I think.
Hello, Mr. Fulmer.
Amplipod.
What time does Sophia come out?
Yeah, they were out and release it. Alex is going to take a video. The bird. No.
Anella, what do you think those little wormy things swimming by were? the transparent ones that were really close to the camera. No, no, the long one. They were like wiggling like a larvation. Not a salp. I know the difference between a worm and a salp.
I didn't see anything.
Oh, okay.
I mean, if they were kind of wiggling like this, they might have been keeping
Oh, okay. Maybe. Well, we did see tons of Ketignaths in the ISIS images, so.
We have thousands of them in the truck.
Oh, okay. I bet it's Ketignaths.
And they are massive.
Wow.
And at least 3 or 4 different species.
Cool. We need to get a ISIS profile.
Seems like we're definitely seeing more things swimming around in this layer than we
What's? Oh, sorry. I thought you were talking to me.
Yeah, I think we're seeing more things swimming around in this layer than in the upper layer.
Yeah, more stuff swimming around for sure. There's more like crustaceans. Look at all these. Oh, look at all these little krills.
And
Maybe.
you're in the krill layer.
Yeah. We suddenly. Oh my gosh. Do you think they just are attracted to the ROV light, or we're actually driving through them?
I think you're in the layer. We definitely got a layer in our nets too.
Oh wow. Oh wow, all of a sudden maybe that must be that must be that scattering layer, right?
I don't think we're quite deep enough for that yet.
The 250?
We're at 250 and the scattering layer is just below that.
Oh, okay.
300 maybe. Maybe even 350.
That was cool. We just like entered a patch of krill all of a sudden. Yeah, now we have 5 minutes left. Yeah, I think those are keating gnats. Those like super transparent little wormies.
Think so.
All right. Can stop. And
Bridge
then
ROV, can we pause and move, please? Bridge ROV, can we pause and move, please?
Copy that.
Thank you, Tony.
so the next steps is 375 meters.
Yep. Going down. Sophia. Find my present.
It's the best part.
Did he fly away?
Not yet.
Still
Still. Thank you.
He looked a little bath. Put some towels in the corner. I definitely should.
That's what it was.
Looks pretty.
Hmm. Oh yeah, look at, oh, you can tell really well that those
This is
are
the
like
worm layer.
Oh, cool. Yeah, well, this is, Anitra said that there was a deep scattering layer like down here somewhere, so maybe we'll be in it. Whoa. Oh, yeah. I guess they're not worms, are they? No.
Well, a worm isn't like kind of everything's a worm.
Yeah, I like to call everything a worm.
It's either a
Kind of a
worm or
catch-all.
fish.
Yeah, wormy.
Worm, fish, or
Worm-like, wormish.
There you go.
Yeah. And Ella, do you know what Kidignath poop looks like? Yeah.
There's also stomach contents in there. I haven't seen like poop personally.
What end does it come out of? The back end? Or the bottom? The top? No, I actually I went on a deep dive a few months ago trying to find out what Kidignath poop looked like. And I could not find anything.
We did. There was a lot of big gas signal in some of the particles.
Oh,
But
well.
I don't remember which ones.
But we don't know if that's
We don't know
coming
if it's
from the
yeah.
Kidignath. Really? Huh. Maybe
Oh,
we'll
wait, no,
see
it
it.
was annelids. Never mind.
Maybe we'll see Kidignath poop in our sediment
We
traps.
probably see it, but
What depth was
we
that?
have no way to
2.
identify it.
That was at like 300 meters. Maybe in our 300-meter trap. There's going to be a lot in our traps.
Yeah. But we're just doing 2 days.
Oh my gosh.
It's probably
All right. Okay. So, oh, beautiful. Salp layer.
Are some of them
So,
ctenophores?
so I'm gonna set that if we can do the same thing. Are we set up for that? Go in the right direction and
Yeah.
everything?
Yeah.
All right. So just, sorry,
Continue.
just drive forward for 15 minutes. Let me set the timer.
I'm going.
Okay, go.
I'm down.
Colleen, did you see the
Oh, it's beautiful. So where are we at? We're at 375. Oh, I see. Oh, sweet. So that was what was that layer, do you think? Oh, well.
Can we just put this over there for you? So you can see?
Well,
I mean, if
Oh,
it
you
was,
have it there.
if it was
do
fish,
I? Oh, right here.
oh. If it was fish, we wouldn't have seen them.
So right now, is this now or yeah, this is now?
That's now, yeah.
But jellies don't get picked up very well.
Right, that's us on the move again.
That's at almost 300 meters. Oh yes. So we're in that deep scattering
Okay,
layer.
Sorry. But we're not really seeing types, so.
And it's only the second.
Do salps scatter? No.
Put in a.
What scatters?
What station?
She was saying of jellies, it's only the siphonophores.
Like,
Why don't you give me a phone?
Yeah. Oh, yeah. So. maybe
What else
it's
scatters?
Stuff that's swimming away from us.
What was your report?
oh, beautiful.
She said it salsa or tuna?
I think there's a Tina for.
And full.
We got a lot of gorillas in there.
Cool.
Huge pyrozoan. I saw some of it. Like it's like.
Andy was just quoting one of my favorite authors.
Bedtime
Oh.
stories for RV pilots.
What's scattering down here then? Can you do your ESP with the animals, Anella, and have them report back?
What are you?
Oh, there's a nice something.
Should I bring it?
There's a lot of jellies down here.
Okay.
Yeah. Come.
Thank you. It's an otter.
Oh, cool. I just noticed the chat up there.
I don't know. Kind of like nope, there I found it.
A lot of salps.
Wiggle.
Yeah. That's it. Okay. Those Keating gnats must be really large, right? For it to be,
Like this, right?
yeah, is
That's
that,
true. That's pretty big.
well, By my standards, that's big. Ooh,
So,
yeah.
I just like the lids. I'm not very good at telling them apart.
I think they're salps. I think the doliolids have like these like rings around them.
Yeah.
We haven't seen any larvations, but we're only at 375. So
And
maybe.
where are they the most dense?
I think there's different species at different depths, but they're kind of. I feel like I've seen them at all depths.
Oh, that was a good unicorn, the bottom view. I'm surprised we also haven't seen any siphonophores.
See this thing? Is that a little fish or something? It's like shiny. Yeah.
There's a siphonophore now that I saw.
Is it? Is that a siphonophore?
I think so.
We're in Salpland. We need 5 more minutes.
Oh,
Oh, squid.
squid.
Oh yeah, squid.
Hold on. See him?
Oh, nice. Yeah.
See that?
Maybe it's squid. Maybe that's what the scatter there.
Oh. Do squid scatter? Yeah, they scatter, right?
Yeah.
Maybe, yeah.
They pulled up some of them in the net, but they wouldn't have gotten the big ones, I guess. I got one like pretty big glass squid.
Oh, they got squid?
Mm-hmm.
Cool.
A lot of little ones and one bigger one.
So I guess Anella was talking to Chris yesterday about possible ways to kind of calibrate how much volume we might be, or like the image area. And Chris thought we might be able to use the lasers to estimate a distance, but I don't know what the lasers are going to hit. But if you go across, like if we did like a grid with the lasers across, maybe we could estimate.
I don't know.
Engage the lasers.
The lasers.
Oh,
just,
maybe not yet. Don't
When we
do
stop.
it yet.
Okay.
Don't do
The
it yet.
lasers just go away off in a point as far as the light will reach.
Yeah,
So I mean. it's
yeah,
hard to
I know,
tell.
because then you don't know, because if it's not hitting anything.
No, it'll just
Oh, it'll
shine
hit.
off.
We'll see it.
It'll
Okay.
hit, but
Yeah,
it'll-
when we stop, we'll turn them on. You can see.
Okay.
it'll scatter against all these little particles, but that's about all you'll see.
Well, I guess I'm not. I don't know exactly what they were thinking, but we still have some time to figure that out if it's possible.
Is this for, so you're wanting to see for the size of things or for the area that we're looking at?
Well, I mean, this
Volume of
transect
water.
is, the point of this transect is to quantify, is to count the abundance of the animals in this layer. So we want to normalize it to the amount of area, like volume, that we sampled. So we're going to need to come up with some kind of an estimate.
Estimate how far the light reaches, I guess. Because you're only seeing what's the light's hitting. And
Yeah.
then past that
Yeah.
Well, I mean, the field of
how
view.
the field of view.
You do the field of view that's
Yeah.
10 foot in front of the sub,
Yeah.
and then make a
Oops.
circle around that. And that's your pipeline. So whatever went through that pipeline is your volume. I'll stop.
Yep, that's it. So let's
Also
see.
up in that move bridge.
The next step is 500 meters.
Okay. So I'm gonna check it out.
Whenever you're ready to.
And you know what? I wonder if you're even gonna see the lasers.
Barely.
See the
Oh,
little
yeah.
green lines.
Yeah,
Maybe
yeah.
They will go forever.
Yeah. There's not a lot for them to hit.
No.
Well, I'm not sure what. Maybe Anella or Chris can
Oh,
say
yeah.
what they were thinking at some point. We don't have to do it right now. I don't think so.
And, yeah, I think you would just be estimating.
Maybe they could compare it to similar camera settings they've done during benthic dives with lasers.
You were doing the
Kind
part
of
right?
Oh,
What's that?
Eyes away.
Well,
The
the problem is just.
Oh,
Well, I'm
it's
not
gonna
sure.
adjust it.
Yeah.
Yeah, I mean, I would
Oh,
say
squid,
squid,
nav
yeah.
camera.
Yeah.
You estimate what you're seeing. Yes, I don't know how you can do that.
Because normally you look at like the laser width against something to estimate
Yeah.
how far, but
Yeah.
The laser width is 10 centimeters, but it's
Right.
only when it's shining on a.
Yeah, and we're seeing
Yeah.
Flat
a
surface,
3D
you know what I
volume that's like a cone almost, right? So.
but if you can figure out from benthic stuff what the normal area is and then compare it to this.
Ooh.
Is it right if I move the camera?
Yeah.
I'm just gonna tilt down.
Yeah. Yeah, it's fine. Is whenever we're not doing the transect, you can change whatever.
Going down to 500. So we go down. So that's zoomed all the way out. So that's basically right there is your distance across.
Oh,
Right?
okay.
That many boxes.
Yeah.
So
I wonder if
if you
that,
just make that go away, you know what that is.
right. We know that
There's
at
your-
that
there's
distance
your
from the
area.
camera what that distance is, and maybe
Yeah.
there's some calibration we can do there. All right.
They'll give you a rough guess.
Mm-hmm. Yeah, that's a good idea. We're seeing different kinds of jellies down here.
Which thermometer? Alright. We're always gonna be doing this way. We're always gonna be attacking this way anyway, so it should be good for indefinitely.
Say so. And like with this one here, you see
Yeah.
that little bit of, see that right there?
Yeah,
That's that,
I see.
right? So
Yep.
now you know, and that camera's fixed in the middle of the cell.
Right. So you know the distance.
The distance from there.
Yeah.
Right.
So if you
this
can-
is a bigger field of view
Right.
than this one.
Okay.
Probably because of the brains I have.
Yeah.
But you could do a comparison across the two, and refine your volume.
Yeah. I feel like this one looks nicer than that one. Is it just because that's a nicer screen?
No, they're actually the same camera. That one probably hasn't done the right balance.
Yeah.
Or I just mean the.
The lights will be
The lights are different.
positioned for that camera as well.
I see. Yeah.
But then you can see the difference between that monitor and this monitor as well. Those two are the same picture.
Oh, okay.
That's a different camera. It's maybe just that monitor.
Yeah, it's just that monitor is different, right?
It's going through the broadcasting system. This is a raw feed.
Oh, I see.
Straight out of the media converter.
I see.
It comes up and goes right to there.
Ah,
This
I
is
see.
going through
This
a
is
switcher.
not as high def, maybe? Or
It
no?
should be.
Oh, okay.
It's just slightly tweaked a little bit.
Yep. Oh, I think that was a larvation house.
Yeah.
I think that just exploded.
I've seen like a couple of those.
Yeah.
Pretty good at exploding.
I've seen like more small fish, it seems like, and stuff.
Mm. Is this is this also? Oh no, 500 doesn't have a deep scattering layer. Okay, so how do you feel about this heading? Ready to start?
Yep.
Okay.
Big siphon at the bottom.
Let me set the timer.
Ooh, look at that edge. Fit. Oh yeah, good.
Oh, wow.
Ooh, nice siphonophore.
Yeah. There it is. Wow.
Okay, starting the timer. Oh, that's larvation, right? Yeah.
That might have been a live one now.
Yeah, that looked alive. Yeah,
Oh, wow,
those
okay.
are, we're in the larvation layer
I've
now.
just seen it. It was so
Cool.
cute. That's really cool though.
Yeah.
I mean, it could be cool to try to collect some of those discarded houses.
Yeah, the one, yeah. If we see some, we'll see where we get like
Not even on this side necessarily.
yeah,
Just
on the, we totally could.
because that's something that we're not going to be able to get from the net, like in
Yeah.
a good condition with the microbial community intact.
Yeah. Yeah. That's a good idea. Ketignath. So many ketignaths in the water.
And like throughout all the depths.
Yeah. They're
Like the
so
top was,
big.
like-
Oh, that's just like a larvation
Yeah,
hanging
a lot.
out.
Must have just discovered it.
Yeah.
Really good.
Oh, what's that?
There's a lot of plants too down there.
It's not as fluffy as like when we were up at like 125. It was super fluffy.
But I think our 500-meter traps are going to be funny.
Yeah, I don't think they'll be overwhelmed though. You know what I mean? Like
Yeah.
this is. I think the 2-day deployment is going to be good. Ooh, look at that guy. Oh, there's a little fish, right? They're like reflecting the light.
We can carry on that move, Bridge.
Probably that.
Huh?
also been a lot more cone bellies deeper down.
Hmm.
It's been jelly land of salps of higher now at like 500 meters and it's not like hundreds. Hiker and oh, beaver pellets? Oh, I missed that. They make beaver pellets. Oh, we want that. We want that too. We also see a lot of feeding nets.
Yeah, I'm just asking if they can preserve some for DNA.
The
Anella
fecal
said they got,
pellets?
yeah, well, or just like the animals or like their guts or something
Yeah,
because,
so I made sure that salps and then are on the list for gut content. or,
Great.
Like gut tracks.
yeah, and then there's so many keating gnats. I think we should try to get keating gnats because they're gonna, I feel like they're gonna be in our sediment, our sediment traps.
Yeah. It'd be good to see. I wonder if they come up alive, the feeding nets.
Yeah, I don't know.
Now that only since we got to the ski plan, we've seen a few houses.
Ooh, I think that,
We might be there, but they're so hard to collect. Like, you can see that all the time. They're just like so delicate. Like there's another one. Yeah.
oh, beautiful. I think those white blobs with the bright white blobs
Mhm.
are Oridaria, which is like a type of radiolarian or Rhizaria. Because we were collecting those with Natalia. I could be, I might be wrong, but Oridaria. It's like it's a protist. It's a single cell.
I don't think this one is right. ORA.
I think Oro, AORO. Yeah, DARI.
Oh yeah, I see. Oh yeah.
Because we collected some of those. And they don't usually come up in like usually if you get them in a net, they're kind of broken. But if you collect them with the ROV, then you get this delicate structure.
Yeah.
Big and up there off to the side.
What's that?
I don't know.
Oh,
Looks like it's a
Out of view.
for.
yeah. We can't move. We can't stop for it. On the way up, what the plan is, we'll use whatever time we have left on the way up to collect.
What?
Also, because we don't want to bring shallow organisms deeper. I could so many of those. I wonder if that's really what it is.
You know, there's a filter tube going down. And then instead
Be
of
wrong.
attaching it to cardboard, you just put it, you use a connector to attach it to the hose that's coming from that seawater. Yeah, in the faucet, and then you just turn it to the right amount that like it shoots through a lot faster. It needed like, you know, you gotta finagle a little with the flow, but it's like 10 times as fast. Exactly.
Right.
And I put the filter back in the filter box. The like box that says filter. Yeah.
Let's see, how much time do we got left? 4 minutes. And Ella says they can sacrifice a few keating gnats. Oh, that's a nice something. Hmm.
Do you think that, like that thing that just went by, you think that's a video right?
Oh, I didn't see it. I'll try to point one out if I see one. Yeah. Powerful.
We can all stop on that move bridge.
Cover that. I'm gonna go first.
Two minutes. I hear that sigh. This is too easy.
It's all easy.
Oh yeah.
This thing is getting a lot more stuff.
Oh, that guy.
Everyone's like,
Oh, salps, right? No. What was
oh,
that?
what's
Was that a salp chain?
Actually, yeah.
Like with the little baby clones? But I don't know. It kind of went by fast.
But I think most of the strings have been sites. There's like a lot
Larvation.
of larvation houses.
Yeah.
Yeah. So delicate, which is why it'd be nice to flip them here, because they're big in the neck, but also they're really, really tricky.
And Ella said we can collect whatever we want.
Cool. I'm just trying to think of things that aren't represented
Yeah,
in the net.
totally. Or if Anitra wants something in like nicer shape than what
It's-
was
yeah.
in the net.
It seems like siphonophores and larvation houses would be good targets. Because I kind of snooped around and I
Mhm.
didn't see any like intact looking siphonophores.
Yeah.
So faunifer is probably going to be quite difficult to keep intact, I'd
Mhm.
imagine.
It doesn't need to be the whole thing intact, but just like, you know, even if it's just a section of it.
Yeah.
Well, it's just going through the hose.
Yeah, well, we'd have to try probably getting it intact would be the best way in the jelly jar, but then
That's-
because they're long,
Yeah.
got to be quite accurate. Plus, sometimes they just see a bit of daylight and then they decide that they're
Yeah.
exploding themselves.
Yeah, no,
Sometimes you don't even get close, and
Yeah.
it's like, oh,
Yeah.
just
We could try and then if
We'll
it doesn't
try,
work, it
for
doesn't
sure.
work.
So the next, okay, so that's, this step is done. Then we go down to 625.
625.
send it.
Yeah, I think we do tuniforts, but they do have some good looking tuniforts. At least like Baroque.
Go ahead.
I think it's just that it's getting too close to the sectors. I don't think it's actually the light.
Yeah, those you can get those larvation housings usually like quite well in the picture and stuff, but
Yeah.
they do go
I
pretty
mean,
easy with a bit of twisting. We
yeah.
could catch one in that jelly jars again.
I think we might
If
want
we
to try that.
if we do that though, they're gonna they're gonna get destroyed in the jelly jar.
That's okay.
At least the unless we find a sinker, you know, one that's
Yeah.
abandoned and full.
But then
like-
we could just filter all the water from the jar, yeah?
Yeah, we can
Analyze the
decide
water, get
what we
the
want to.
depth, and
Yeah.
Because I'm interested in the microbiome on
Oh, yeah.
the elevation houses.
Yeah. We can do chemical analysis of the water.
Mhm.
Oh, nice.
Yeah.
Problem solved.
Because Colleen, when you caught them before, you just
Well,
filtered the water, right? Were you, like,
Well, we collected the sinking houses, so they're
Yeah.
pretty goopy.
but
and
the
they
samples
just-
were filters.
Yeah. Well, we don't filter the whole sampler. It's you can just slurp up the goop.
Okay.
'Cause
And
it's
you
all
just- you
stuck
just put it on the
together.
filter to make it easier.
Yeah, I did. We didn't have to put it on a filter. I just did because I don't know why. Oh, because there was too much liquid. There's too much liquid, so.
Yeah, that makes sense.
But if it's a fresh larvation, I mean, we can do it. We can try if we have time.
Yeah. And if there's not other priorities.
Disease control.
Colleen, do you feel like the
So
amount
it's
of
a
particulate
piggyback
matter is
off
diminishing as we go
what is
deeper?
there.
It's definitely
You
diminishing.
just put
The
it in
surface
parallel.
or the shallowest ones
Said that
had
like
like
my first
beautiful,
check, like,
fluffy,
Parallel
so
wire,
much marine
everything that's
fluff.
gone off in
Like
here,
it looked like post
over at
bloom
chocolate block, and then here.
something. But yeah, the particles are getting
Done. Is that right?
smaller and denser.
Don't know why.
And you said we're right in the cortical bloom right here.
There's
You
yellow
see
blue
it,
issue
and
above us.
Where are we going to?
Ooh.
Yeah.
625. Yeah, we still have a little ways.
Take
I'll
over.
try this.
Yeah.
Yeah, 65.
65 up.
Hmm? I'll keep you like that.
How small of a can we see with these cameras?
Pardon?
Uh,
How small
I
particle
think
can we see?
Pretty small. I mean, we can zoom in quite a bit, but it's getting it in focus in the midwater is the hard thing because obviously they're floating in and out, in and out. They're not staying at a steady place where the autofocus is able to actually keep them in focus the whole time. So then we have to try and do it manually. But I mean, we've got two things that are floating independent of each other. It's kind of difficult
Yeah.
to.
No, but just on the screen as they're passing by.
Well,
Like
we
that,
can- we can
this
zoom.
big chunk is probably like, that big.
A
The,
centimeter.
yeah, the ones that are in focus, like right when they get up to the camera, are like that big,
That's about
like
centimeter.
a centimeter, or maybe a little bigger.
Everything looks bigger on the camera.
Yeah,
Do you
yeah,
have the lasers?
But the lasers just shine off into the distance.
yeah.
The
They're not. They're too
particles we're
small.
seeing are probably quite small. Right? They can really
Yeah,
the small
yeah.
I mean, you
particles,
can
like,
see them up
just the
here, and you
dots,
have a
like,
reference
nothing
to like the size of
Maybe
it and
there?
stuff.
What do you think?
I'm trying to remember, like, cause we did a lot of work with the Phaeodarians, which are like this big, so I guess like a centimeter, and then or so, and those. I guess we can see things that are smaller than that. It's really hard to identify. Like, yeah, I guess we can see particles that are smaller than that, but in terms of like seeing nice identifying stuff,
Yeah, I'm just thinking about the particulate organic matter that's just sort of that look like the marine snow or the specks of white.
maybe it's like 500 microns and bigger. Maybe that's a good guess.
Just thinking about it in terms of my filter pore size when I'm collecting from a CTV bottle.
Yeah.
You know, I don't generally think of the particles as being very big.
I would guess.
Isn't it funny, like,
I would just guess.
it comes out of the CTD like nothing's in it,
Looks like
and then you
nothing's
look
in the
in
water.
here and there's so much stuff.
Exactly.
Like the CTD, it looks like there's
625.
nothing in the water. And
Great.
So
here
it's
it's like
confusing.
Okay, so
full of things.
Yeah. But yeah,
Whenever
we're
you're in a
listening, like,
heading-
pretty far, I guess.
Yeah.
like hopefully going into the current,
It's a wedge depth of field.
But
um,
it's still expensive to always
then
to me.
you can just start going.
Go ahead.
All right.
Also,
Also,
most of my working experience is in oligotropic gyre, so like a couple of
Yeah, it seems like a
dives
lot.
at Embarios. I'm like,
here we go.
It seems very rich and
So we're at 625. Oh, nice. Same. So what time it is? It is 9:20. Wait, what time is it? 8:20.
I was gonna say 9:20.
Yeah,
Right.
sorry. I have terrible vision.
Oh,
Yeah. Allegedly.
I think that's the download piece one. Yeah, I think that's just the section.
Oh,
But yeah, I think
cool.
that's the one we're using for transects. I think it's this section.
Hmm?
That's a siphonophore, right?
Yeah.
That okay. Oh, those were Phaedarians. Did you see that those three things stuck together? That was those were Phaedarians. Not the same kind that we were studying, but different. Yeah, they're protists.
Should we explain for the chat? The viewers?
Okay, Bridge, we can resume that move.
On the move now.
Oh shoot! Wait, timer. Oh good, the timer is going. Oh, I was worried. Yeah. Yeah, there's way less particles compared to shallower.
It's still quite a lot.
Huh? Yeah.
It's a lot to me.
When they're smaller.
Yeah.
I'm not used to one every day. So.
Yeah, totally. For being in the middle of the ocean, seems like a lot. Yeah,
Although this
it's a
stuff
huge
would be
phytoplankton bloom here.
from
Oh, beautiful.
days and weeks ago.
Yeah. From a long distance away.
Yeah.
What's that one? Yeah, check. That one will just put your head across that one.
It's a good one.
Oh, yeah, cool.
They've got some
I don't remember if I asked you this or if this was just in my head and I didn't say it out loud. Our camera settings are on manual, right?
Yes, they are.
Great. I don't know how to read all your screens.
No, I'm just pointing out. So that bottom one over there that says size Ulysses 70. That
Oh
one?
yeah.
See how that's all just gray?
Yeah. Okay.
All manual.
Good.
Yeah.
Great.
So then, yeah.
How did you record the same thing for using?
We could maybe we should take a photo.
You can do that if you want.
Yeah.
Well, so it's the bottom right.
Yeah.
Maybe let's write them down because that was a bad photo.
Well, yeah.
Wait.
I can read it out to you if you want.
Sure. Okay.
Squid.
Yeah.
Oh yeah. All the cool things are on the camera. The nonsense. Oh, there it goes. Yeah. Cool.
So ISO is 640.
So 640
Shutter speed 1/60.
shutter 1/60.
The like focus is half in between far and near.
It's the midpoint.
That one
That
will
is-
always be there.
Oh, okay.
Oh, okay.
Yeah. So
Is
that's
there anything
more
else
about
that's
control.
relevant?
I mean, the iris, but we don't know
We
the
don't have
iris is.
control.
Okay.
Yeah.
Yeah.
So we don't have the iris.
Well, I don't know if the iris has been set all the way open before they've set the ISO at 640.
I believe it's open.
Yeah, all the way.
Yeah.
So
Iris all
it should,
open.
it should be open all the way. Yeah.
Yeah. We were here. We said it.
So iris was open all the way, and then the ISO was set to 640.
Yeah.
Yeah.
Okay.
And then on the left, is that what lights are
All
on?
open.
Yeah.
I'll take a picture of that.
Yeah. So we just have not the. All
to
I
make
think
sure.
all the forward lights are on.
Just to make sure it can be recreated.
All forward lights are on. Okay. All right.
Yeah, all of them are on the first one.
Oh, somebody asked, where are you at this moment? We are basically west from Puerto Madryn in Argentina, but pretty far offshore, like 3 days steam.
We're east.
Oh, did I say west? Jeez. East, yeah. We're not on land. It was about a 3-day steam, but we're pretty far off the shelf. We're very far off the shelf break, but in this pretty big chlorophyll patch. You can see it from satellite. It's a huge region where there's high chlorophyll. And so it's really far offshore in the open ocean. You should be able to see it online. I think there's probably a map. Is there a map online of where the Falcor is?
Yeah.
Yeah.
What was it?
The website.
Yeah, the website link. I put into the chat.
And then this cruise, this cruise we want to cross different ecosystems. So right now we're in a relatively high productive, more influenced by the subantarctic waters, lots of life, phytoplankton blooms. We're gonna move north, and as we get north, we'll get into more low nutrient waters, which are called oligotrophic waters, and so there won't be the phytoplankton in the surface won't be as productive, not as large of blooms, and so we expect the also, you know, at the same time, the deep ocean has a different ecological province. So we want to go across these different types of ecosystems, and then we're going to get offshore of Rio de Janeiro in Brazil, and that will be a different ecological province. So we're trying to observe these contrasting ecosystems and understand what's going on with the animals in the midwater, these mesopelagic animals that are a big part of the carbon cycle. All these particles in the water, these are sinking carbon away from the surface and into the deep ocean, but their fate is kind of determined by all these animals living here. So we're trying to understand how the animals are controlling sinking particles, basically, and especially what's happening inside of their guts when they digest things and when they poop things out. I mean, a lot of these particles are fecal material from animals. So what happens inside of their guts? At least that's our hypothesis. What happens inside of their guts is gonna be an important part of how much carbon gets sequestered into the deep ocean, and we think we want to find out if that changes as we go into different ocean ecosystems. So right now we're in a pretty productive area. How much time do we got here? We have 3 more minutes at this depth. Oh, I think someone's asking, how deep can Sebastian go?
4,500.
Oh, what?
4,500 meters.
Okay, 4,500 meters. We're only going to 1,500 meters though, on this dive.
Does the live stream see the downward facing HD camera as well?
Yep.
I was just curious because I feel like we can see it.
Well, that was a nice something. Gelatinous.
There's still so many kitty cats.
So many ketid maths.
I'm really curious. if
It's
we're gonna
wild.
See their signature in the particles.
And a lot of depths.
Yeah, like the whole time.
I mean, I haven't really thought about ketid maths before. Not really.
Same.
They seem really important though.
Backtrack.
Oh, no, thank you. Sure. Oh, yes, thank you. Maybe that's the button.
It's the hose.
Yeah, it's sleeping.
Oh, you have your own.
I'm on the boat.
Hmm.
You were saying that you see a lot of, like earlier y'all were talking about animals that you see off of the California coast. What do you normally see out there?
You actually
I've
know.
always gone on a couple dives there.
She's a massive teacher.
But
I'm like,
it's just like a lot. There's just like a lot of jellies, a lot of ctenophores and saxonophores, larvaceans. We get these like giant larvaceans. Much bigger than normals. Yeah, Colleen is more familiar with that. Most of mine has been.
Because I grew up in California, but I've never like done the road here up there. So
Yeah.
I was like,
Where did you go?
in the Bay Area. So with Andy.
Yeah. I went to school in Santa Barbara.
Oh, did you?
Yeah.
That's awesome.
Santa Barbara City College. Marine technology.
Anyway.
Cool.
Hello. Muchas gracias, amigo.
De nada.
Ian.
I mean, I'll go do it myself.
You have to wear it, is it?
In
your office?
No, don't worry about it. I got it.
Hey, Sophia,
Yeah.
or Ian, is there a reason why we're still in the SBL position?
It was because there was SOI issues. What do you think? You can stop all
Yeah.
the
Okay. Just curious. Thank you.
It was working fine. It was just throwing us an alarm.
All right,
Okay.
that's 15 minutes here.
Making some coffee.
All right.
Okay. So the next steps is 750 meters.
750.
Yeah,
Going down.
going down.
So are these the, is this like the top and bottom calling of all the net depths?
Let me
or,
read
like,
Anela's
midpoints?
notes. Thus, we propose to conduct ROV transects at each of the edges and midpoints of our mock nest. So it's both,
Edges and midpoints.
yeah, it's
Right,
kind
so
of
it's
the
like 10.
boundaries of where they're sampling with the nets.
Look at that big squid.
Wow.
Big squid.
Do you get Humboldt squid over here, or is that just the Pacific?
Mm.
We haven't seen it.
How long has the Buckwalk only been the Atlantic so far?
No.
We were in
when I
Chile for a
start-
long time, right?
Yeah,
Oh,
we
yeah.
started in Costa
Long
Rica
coastline.
and then went down out to Rapa Nui back and then down all along in the fjords
Oh, then went around.
around Chile and then all the way down and around.
That was sort of fun.
Down to Antarctica, back,
Okay, cool.
South Shetland Islands, back, and then up, and down, and up, and now. Yeah, it's been a lot, well, about a whole year, basically.
Yeah,
right?
more than a year.
More than a year and a half,
Wow.
probably.
Well, so when the ship got out of the shipyard in Spain, did it just go straight to Costa Rica?
No, it went
No.
across and did.
Did 2 trips out of
Puerto Rico.
Puerto Rico, yeah.
Okay. And then did you go through the Panama Canal?
Yeah.
Oh, cool.
Did you do ROV dives in Antarctica? Was it summer?
Hmm?
Was it summertime?
Oh yeah.
So it was probably a lot of stuff in the water.
No, actually there wasn't that much. Yeah, we one scientist was studying krill and we weren't getting it. we weren't hitting the spots.
We can stop that move, Bridge.
Yeah, that's me already all stopped.
Mm-hmm.
Okay, roger that. Just checking. Thanks. I got it.
We went 2 locations. One was down kind of peninsula. Just started to go around there, and then the ice stopped us. So just kind of meandered around there, and then went a little east. The other time we went down around to the western side of the peninsula. And, how far south we got? 75 degrees. It was quite a ways.
Did you go into port anywhere in Antarctica, or did you
No.
you went to Punta Arenas?
Yeah. Where was that?
7 feet.
There's a lot of squid down here.
I went on vacation to Chile before this cruise.
What about?
We went to Punta Arenas and the Torres del Paine.
Oh yeah, I did that. I
Yeah.
was there. Yeah.
We did that too, yeah.
Yeah.
I think we were on opposite rotations then.
Yeah, it was beautiful.
Yeah, it was good. I enjoyed it.
Yeah.
Where is that place? Puerto Natal is that the one that's
Yeah.
close?
Yeah.
Yeah.
Oh man, I'd go back to
Oh.
Yeah.
Do the W
Extra.
again.
Extra.
Well, we're not that organized.
You have to.
I think that requires a bit of forward planning.
Working in the little cabins and stuff. Did you do the W, or you just went up?
No, I was just we weren't that
Oh, you-
organized. We just did some little day hikes
We
around.
camped.
Yeah.
Did you?
Not
Oh
me.
no, we were. Ooh.
So we were like
Camping, no.
we did the comfort trip.
Camping or glamping?
We were in the cabins. Although it's not very glamorous because you're in a room with like 8 people total. It was like 4 of us.
Yeah, no, me and Lena did tent and then the fancy tents on the ground.
Oh yeah. They're
Fancy
set up
tents
before
above the ground.
They're set
Yeah,
up for
like
you
the,
when you
like
get
the ones
there.
that you set for
Wow. Yeah.
They're basically,
They've got a
you know,
lot
it's
of
like hot
amenities
campers
there.
on, like,
A
beaches
lot
and
more
stuff.
than I
It's basically
expected.
like that, but like on a wooden platform.
It's quite nice.
I remember being on
Yeah.
that hike saying, "Thank God, we're not in one of those tents." I'm like, my knees have given up. I just want to bed right now.
Oh.
It was good though.
Hey, I was in a really comfortable hotel eating really good food, so I thought that was pretty good.
$7.50.
Okay.
I just got a case of hiker's nostalgia. You
Um,
look back at it and be like, oh, it was so amazing. But at the time, you're like, oh, my knees. When?
I was basically.
Okay,
I was
so if you feel
basically
good about
gaslighting my
going
mom
forward,
the
then
whole time.
well,
Just half a mile left. Just half a
Okay.
mile left.
Oops.
And she's
On the
like,
window.
I hate you so
you can go, and I'll just set the timer. Okay.
much.
Going.
Good times.
Wow.
Yeah.
I started the timer.
I think I need to go on another backpacking trip.
Yeah, it was cool. I liked it, but now I'm like able to look back and say that. Pretty sure at the time I came on board. We did it before.
Yeah.
Are we going into the current?
Oh my goodness. Yeah.
Is the current still like half a knot or so? Oh, beautiful.
Yeah.
Yeah.
Okay. Good.
Yeah, because I'm going full step.
Okay. Oh wow. What's that?
That's a scythe.
It is?
Yeah.
Okay. That was cool.
Seems like that's the dominant ones down here. This is gonna be really cool data if we can get it analyzed.
Yeah, I think we can. I was chatting with Anella about it last night. Or I mean, it's certainly
We just have to
it's
figure out
certainly
who's doing it.
possible. We have yeah, I mean, we literally
Oh, is that a
specialize
fish?
in that.
Right? Kinda wiggling around.
Oh, that's a nice
Oh,
larvation.
yeah. You see it was like red. The actual animal.
Oh. Oh, I think there's like red-headed. There's one species that's called like red-headed something. Yeah.
That could be.
Yeah.
That would be great to capture. But seems hard.
Oh, someone asked if we're collecting. Well, we're doing these quantitative transects, just moving forward, straight, horizontal for 15 minutes. And we're doing those at 10 depths down to 1,500 meters. We have a bit of a time constraint on when the dive has to end, just so we can get other operations on the ship going. So we'll just, whatever time we have left after we do these transects down to 1,500 meters, we're gonna ascend slowly and then collect on the way up. And we haven't entirely decided what we want to collect. Kind of, we're seeing what we see while we're doing these transects on the way down, and then we can kind of, obviously we don't know what we're going to see on the way up, but one of the ideas is we have one of the other main things we're doing on this cruise is these mock nest tows, which is this huge net. It's like 10 meters and it goes out for 8 hours, and it's quantitatively sampling animals at discrete depth intervals, so that this huge team on the ship can is sorting and classifying. We're getting all types of samples, DNA, taxonomy, all gut contents, isotopes, all sorts of things from these animals. But this nets sample in a certain way. There's a certain bias when it comes to net sampling, and so we're doing the same depths with the ROV, and that's going to give us a different view of what kind of animals are living at these depths. That's why we have to do these transects. It's kind of boring. We're just moving straight and not stopping, but later we're going to analyze this video, and because it's consistent, it'll be quantitative, and we can actually quantify, count the amount of animals per volume. Oh, that's a beautiful one. And then compare it to what we see in the nets. And it's going to be different because the ROV can see all these delicate things like siphonophores, larvaceans. But there's overlap, obviously. There's a ton of Keating gnats, which I heard they brought up tons of Keating gnats in those nets. But anyway, that's what we're doing. But yeah, we'll collect some things on the way up, hopefully, if we have time. But right now there's like a huge team of scientists working in the lab, sorting through all of the specimens that were collected in the nets. It's a ton. It's so much work. That's why you are stuck with me sitting here, because I'm not in the lab sorting animals.
Neither did I. He's moving.
Oh, beautiful.
You didn't tell him to start moving. I didn't tell him to start moving. You told him to start moving.
He saw us moving. Yeah. Did you ever tell him to stop?
Yeah. He says we've already stopped.
Oh.
So that's a dangerous ground because now we're getting behind the guys a little bit.
750. We can go a different direction.
That's fine.
No, that's right.
Okay.
I just need bridge. Bridge, can you slow down just a little bit? We don't want to get too close to the back of the boat.
Big squid. Big. How about that?
No problem, slowing down.
Oh, I didn't see it. Yeah.
Well, you're smart enough to stay away from the thrusters.
Oh, yeah. Not like some fish. Yeah.
Squid especially, they love the back of the thrusters. They love getting too close to the thrusters.
Oh, look at. Oh, wow.
Oh.
An aft.
Uh-huh.
Hanging out.
They like the light, I think, right?
They also.
There he is.
Wow.
Oh.
Yeah.
Oh,
that's
cool.
That's the cool squid with the
The flashers.
Alacutus.
Oh.
Is it? Yeah.
I think that
I would need to look at it a little closer to for.
Oh.
Ooh.
Uh-oh.
Uh-oh, it was.
Oh.
What did I say?
It was
Yeah.
not that smart.
Why didn't you stop to look at it?
Oh, that was a one. It's not like, see it.
Thing. Yeah, I think we lost it, man. And I still know what it was. I can't remember. Yeah. That was my highlight. Lost it. This is all away. First time I got there.
So docking camera, second time, go, you see the bullet. So on the side, second, first, or that.
I love these particles at this depth. The particles at this depth are cool. Because look, some of them you can see like that they're like stringy. Some of them,
Yeah. They like
yeah. I think that. Oh yeah. Oh wait.
Well, that's a.
Yeah, that's probably us.
That's a tentacle.
Yeah, that's not a.
You're the only one who's more excited about the particles than the animals.
Yeah.
You could let them have it.
Yeah.
Like this guy?
I'm sure.
This guy?
Guy who loves dog puppies and flowers.
Mm.
That's him. As advertised.
Yeah.
Hmm. We might be able to go a tiny bit faster on that, like
You know what?
0.2 to 0.3.
That is full
Oh.
speed.
Oh, really? Because of the current.
Mm-hmm.
Oh, wow.
Yeah.
Then we are not. Oh, look at that copepod.
Let's get a little bit
Okay.
stronger down about here. Margin
Well,
up.
as we go,
Nice.
and
Then that's good. That's perfect then.
yeah, that's everything like maxed out.
Okay. Oh, whoa. Check out that. What was that?
It was a fish, right?
Fish.
Oh, cool. Not allowed to stop though. Let's see how much time we have left. 5 minutes.
And then I'll try isolating like
There's also a really good one.
food issues or like messed around
Guys,
with
been
our
there.
outlets. And then I don't know what I did. And I just started.
Yeah, it's like almost 90% of the fault since you last year these days.
No, but it was like, it wasn't the same, like it wasn't anything like
No.
shit and it was
Ah,
like camera feed after I like put it onto the browser. Or started the broadcast.
Oh, so that's a sinker.
It's fixed, but
That's like an
Yeah.
abandoned larvation house that's sinking.
Mm-hmm. And those are more sturdy usually.
Mhm.
So you're saying.
Yeah. Well, if you collect them, they kind of all, it just all sticks together.
Yeah.
Yeah.
do you want to know like,
Tell me, always.
do you know when you
Mm-hmm.
You can just make it record right there.
Boom. This is one in parallel to the other one. Yeah.
I mean, they're not actually that sturdy. They also
But
break apart.
relatively.
Yes. Relative to the fresh ones. Oh, look, is that another fish?
Yeah.
I think so.
Okay, well, this is where all the fish are. I guess it's daytime, so it makes sense they're hiding
Mhm.
down here.
What's the normal depth that you see in that?
It depends. Like it's just like surface search at night, deeper during the day, but like so like behind you the echogram is showing you kind of like those deep scattering layers. So you can see there's kind of like 2 layers.
There's a big larvation there.
And then out there in the dark.
Oh, wow. Cool.
Whoa. That's a good one.
Nice.
Wish we could stop and get it.
No,
No.
not yet.
So it's going to be the operating procedure for every dive. Oh,
I'm sorry.
Will we
It's
have
amazing.
time to stop for creatures at the end after?
Yeah.
On the way out.
again, that'll be the other guy's ship. You know, lose it off with 12.
It's okay.
That's the goal.
I'll make sure the
This is our- This is just our first one, and then we see how this goes, and then we can shift things. But like these quantitative surveys are going to be
Oh, look at that guy.
super
Whoa.
important.
Two
That's
guys.
awesome.
Whoa,
That's really cool.
nice. Like if we stopped, then it's just like analyzing that would be really, really
Yeah.
in the
Be a
butt.
data nightmare.
And
Yeah.
like, this is really valuable data. Yeah, it's boring.
Really valuable.
for us
You know
right
what?
now.
And like what's so rare is that we get both the Mach ness and the ROV at the same place,
Mhm.
right? And time and
Because they're both biased, so it's good to have them that complement each other.
Yeah.
but yeah. Hopefully we get time to sample. But the other thing is that mock nest needs to go back in the water by 6 p.m. to get the nighttime
And it takes
migration.
a while.
So we need to. Oh no, not 6 p.m. Don't quote me on that time.
Uh-oh.
I don't know. Look at
I
the
don't
schedule.
think that's true.
Look at the schedule, but
As ROV pilot for training, we
we're
have to believe the schedule.
we need to be out. We need to be out of the water by 2 to give us enough time.
6 a.m. Wait, no.
No. I think it's 6 p.m. And then I need to be out of the
Yeah,
water by 2.
It says 5 p.m.
So we have time to do a couple things before that net needs to go in the water because it's about the animal migration. So you have to like do it at the right time so that we get them in their nighttime migration.
You don't want them to like.
That's
migrate
kind of the
halfway.
constraint we have on the time. But there's other days.
Weren't we
I
gonna
don't
do
know.
some night ROV dives?
We want to try to do a night ROV dive.
We're more restricted with times because we need to kind of wait
Yeah.
until we've got an opportunity where there's enough ROV guys on shift
Yeah.
to launch and recover. So it
Well-
would be like launching around about midnight or launching at 6 at
Or midnight, but there's crews overlapping. Someone stays up and helps.
Yeah, but it's more likely to do it when Doug goes off, before he goes off, and then carry on the whole night. But then it would be an extended dive
Yeah.
because we'd have to wait.
But you
until
can't recover until. Yeah. Okay. Well, that's good to know.
We're
Or-
just more
or you-
restricted because it's possible we could
Yeah.
do a handover, but then it's kind of awkward as well because
You're looking at a 6-hour dive or a 12-hour.
Okay, we're done at this. The alarm went off, so that was fast. Maybe they're all blurring together now. Yeah.
okay, we're all
All right.
stopped, Bridge.
So the next step is 875.
Coming to a
Going
stop.
down.
Okay, so that's good to know. So it's either like midnight, maybe,
Well,
or 6.
midnight's not
PM?
ideal.
Oh.
Ideally, it would be because the opportunity for us to have enough guys on shift is when we're actually meant to be handing over. And then if there's any delays, somebody's gonna have to stay out later or come out way
11:30,
earlier.
12:30.
We'd have to be like at the point of doing floats or thereabouts at like
Your window
half 11. Yeah. So it is possible to do, but then probably better would be, I don't know, what's Doug doing, 8 till 8 or 6 till 6? So he's doing 8 till 8, would do 8 o'clock off deck, back on deck, 8 in the morning.
Well, I think he was doing 6.
I don't know
or
if we
6.
can do a 12-hour dive. Like, I don't know if we have enough.
You got 6
Yeah.
hours or 12 hours. So
Yeah, we're kind of
I see.
tied to that.
Then we would probably have to do a 6-hour. Okay, good to know.
It's going to take a bit of planning as well.
It's going to be this fella right here.
But about doing night dives. Look at this guy.
Nice.
It's only 9.
So am I supposed to go back?
Yeah.
Oh, okay. What were you saying about night dives?
It would have to be a 12-hour dive or a 6-hour dive, but it's going to take a bit of coordination beforehand if you want to do a night time.
Yeah,
Good to know.
it will take coordination.
Mm-hmm.
Coordination and planning.
All right, so let's see. We're going to 875 now.
875.
Okay.
Mm-hmm.
Sea beast.
So it is a beast.
It's a large sea beast. No, I think it was one of those worm things.
Oh, yeah.
You know.
You know, the worm lines.
You know the worm things?
Hmm.
There's definitely a lot of more stuff down here. Like less particles, more animals.
It's a nice one.
Yeah. Nice try. Nice try, mate.
We're doing that.
I do want to answer that question about daylight and nighttime. Orange.
Oh. So how are the animals aware? Daytime and nighttime. Oof. I'm not the one to answer that one. I mean, I do think they can detect. They are really sensitive to detecting daylight, so I do think they can see more than we can see. But do you know?
I mean, yeah, my understanding is like usually at least some light penetrates down to like 1,000 meters, and so they have really sensitive eyes. Or
Yeah. I mean, if you're looking up,
yeah,
you can see it change, but like.
So they're not like seeing maybe as good images, but they're able to detect if there's light.
Yeah, I think at least down to 1,000 if they have really sensitive eyes. I don't know about the ones that are even deeper than that. This is definitely an Anella question.
DAVM below 1,000.
I think
Maybe not
this
to
is
1,500.
Yeah, I think there are. Well, only based on my recollections of conversations with Anella about science stuff, but I might be misrepresenting that.
Yeah.
We need her.
As soon as you talk about
It's 75.
It's just like spiking all the time on that.
Oh,
Yeah.
that's good. 75.
Okay, ready to go?
Always.
All right, let's do it.
Engage.
Yeah, but I think they do DVM based on light time.
You know,
That's true. So they might start migrating before that. I started this other cool study a while ago. They did during a solar eclipse. They found that they started going up. So that's how they determined that it's based on light for most of them.
Oh,
They did go during up. They did go up. So like, and they also, I think the migration changes on like cloudy days versus sunny days.
Yeah.
So it's like it is based on light levels.
oh, and then the other thing is, I remember Kelly from Ambari, like they had a cool study where it's like once the predators showed up, the prey started migrating.
Right, 'cause some
So
of
they
them
were kind
do-
of like responding to their predators, like to
Yeah.
escape. So
Some of them do like a reverse migration where they actually go deeper at night to avoid the predators.
yeah,
for
and
most
then
part,
Yeah, maybe some have just like an internal clock.
Yeah.
Juvenile colossus.
Well, whoever asked that question, sorry, the super experts are not here in the room.
Sorry, I shouldn't have prompted you. Ooh, look at that. It's a good compliment.
Oh, beautiful. Oh, yeah. Whoa, there's like a lot more big
Actually,
animals at this
that
depth,
orange
right?
one, I think they've got a couple of those in the nets.
Yeah.
They're these
Oh,
like
it-
low bait
yeah.
herbivores.
Stuck there.
Got someone in that jelly jar without deviating from the course. That's true mastery.
Right in the club. Trying to hold on.
What's that? I got. Is that a fish? That thing? It kind of It kind of was very reflective. Yeah. Hmm.
That's just how it goes.
Hmm.
I'm sorry, I just thought this.
Oh, that was a fish. I think that was a fish. Maybe I imagined it. I'm hallucinating. Five minutes. Someone asked how deep we're going. The deepest depth we're going is 1,500 meters. I think the seafloor is much deeper, probably closer to like 4,000 meters here. Oh, deeper?
Was it 5.5?
Oh, 5.5? It's 500
for a while.
Yeah, it's like 5,500 meters deep here, so that's really deep. But we're focusing on the midwater or the mesopelagic. So we are going down to 1,500 meters and focusing on pelagic animals. The reason being that, like, this is pretty much a black box in terms of understanding deep ocean ecosystems and interacting with the carbon cycle. It's so complex and hard to observe and study, and you know, most carbon cycle, like our understanding of the carbon cycle in the ocean is not very well defined in this region of the ocean because so many ecosystem interactions and all of those things determine what actually reaches the deep ocean all the way to the seafloor. And so we're trying to figure out how to make those observations in new creative ways so that we can start to figure out what's controlling how much carbon reaches the deep ocean. And by carbon, I mean all this white glowing particles, the marine snow that's sinking. And so that's why we're focusing on the pelagic, because it's so, the processes happening here are just really not understood well enough to do things like put them into a carbon cycle model or even like an ecosystem model. Yeah, so we're not going to the seafloor.
So right now we are at about 3,500 meters. So we shallower than it was yesterday.
Oh, okay. Well, Anitra says we're 3,500 meters is the max depth here, but we're not. The ROV is just going to 1,500.
Well, oh, do you guys have some?
I think they do have some. Oh, no. Somewhere in the boxes.
Okay, how much time have we got? 2 minutes.
Yeah, sure. Because I'm not doing that. Yeah. I would like to pump water.
Hmm?
The last question.
How about how deep we'll go?
No, why we're not stopping this issue.
Where's that question?
The last one.
Okay, why you're not stopping to zoom? Oh, someone asked why we're not stopping to zoom, because right now we're doing these really important quantitative surveys. So we're driving horizontal at the same depth for 15 minutes at the same speed, and this video that we're collecting is quantitative. We have the same area that we're imaging, and then this video will go through image processing, and we will be able to quantify how many animals we saw in this volume of water, and that then we can actually say how many animals and what species are at this depth. And so that's why, I mean, yeah, we're not stopping and zooming, but the video exists, and you can freeze the frame and extract the animal image later and identify it. So it might not be that satisfying for us right now, because we just get these brief glimpses of what it is, but later the data will be way more quantitative, and in the long term more valuable in terms of quantifying the amount of animals. We will stop at the end of at the last half of this dive. We will stop and zoom on animals and collect. Yeah, so that was the end of 15 minutes at this depth. So now we can go down to the next depth, which is 1,000 meters. We're going to do this all the way down to 1,500, and then with the time we have remaining, we're going to slowly ascend, and that's when we'll stop and zoom and look at things and decide if we want to collect them.
What was that?
Have you been logging the transects right now? Okay, that's okay.
I've made the button though.
I saw that. Yeah, that's why I asked. So
we could start doing that.
So,
yeah.
But do we need to put in a late entry for that one?
Well,
Started a while ago.
Well, I don't know. I didn't record the exact times.
Really?
I haven't been recording the times.
Oh,
I mean, it's recorded in the video, so we can get it
that's
again.
true. And I guess your audio was. But for the maybe for the next one.
Yeah, we can.
Yeah.
Let's
But
do
I'll
it for the
definitely
next
record
one. But
when we sample.
We can get that data from the video
Mhm.
and from the ROV. So
And we're ready to log biological samples like Niskin models.
great. But yeah, let's start logging the transects too.
Okay. Are we on now?
No. Oh, Anitra and Ingalls is chatting with you.
Oh, that's my sister. I was gonna say.
Oh. Sisters. Yeah, my family is all sleeping right now.
My sister's in New York City, so
Yeah,
it's not that early there.
yeah. Oh, someone's asking if we have the jelly blender. No, I know exactly what you're talking about. That is part of a project that my colleagues at Mbari, their collaborators, are working on. I think they're still working on that. We are not using that on this cruise. I'm personally thankful. I don't want to see that. But I guess it's good for their project. Yeah, I remember the first time I saw them presenting that at Ocean Sciences, and I feel like I needed like a warning before they showed that video. No, we're just slurping. So we have the D-samplers or jelly jars. I'm not sure what their official name is. Yeah, at Mbari they're called D-samplers, but I've heard them also called jelly jars. So we're using those, and then we have the suction sampler.
There's a fish fry.
De nada.
We could, if any of someone's asking if we can talk in Spanish. Unfortunately, I'm sorry.
We have
You don't want to hear me talk in Spanish, but we do have a lot. Yes,
people in the science party that are Spanish speakers and Portuguese as well, and they will be coming in for our dives when they are available. We had a net tow come up earlier today, and they are really busy in the lab sorting animals. So none of them are here right now.
Yeah, we do have we have people from Argentina and Brazil. They're really busy sampling animals right now from the net, so we're hoping that they can come and kind of narrate for the Spanish and Portuguese speakers.
Ready?
Are we done? Oh, it went off? It was just like
1,000
so.
meters.
Oh, we're at 1,000 meters. Sorry. I'm glad somebody's
So calling
paying
let me know when you
attention.
start.
Yeah.
Hit the button.
Okay, so we're good to go.
Alrighty.
We have science
Yeah.
from Chile,
Engage.
from Argentina, and Brazil. I'm noticing someone in our audience is from Chile.
Yeah, we have Chileans on board. Yeah, Ruben from Concepcion.
Been storming.
What?
Been storming.
I like when the kid gas try to like outrun the road.
We have an artist at sea here too, also who's from Ecuador.
Mm. Oh, Paula's from Ecuador?
Yeah. She's been living in Buenos Aires for a while.
Oh, okay.
Just moved away. I'm still amazed at how many particles are down here at 1,000 meters.
The Niskin bottle gives you a really kind of specific view of the world.
There's a lot of fish down here.
Yeah.
They're like the vertical orientation. Holly, we're going to 1,500, right?
Yeah. We have the next one will be 1250 and then 1500. So we just have two more after this. Do you plan on sampling ctenophores? Maybe.
We can.
We have,
Yeah. Yeah.
yeah, our goal, so we do have some people interested in animal evolution and sequencing genomes, and then some people interested in animal physiology, and then more of the people on board, and kind of like definitely the primary goal of this project is thinking about the animal guts, and so we definitely want both the fish and invertebrates. So yeah, ctenophores are probably really important. So if we can sample them, and we would probably focus on sampling, I mean, their guts. I don't know. They kind of have guts, right? They
So,
kind of
yeah,
digest on the
they
outside,
do
right?
have a mouth and a butt.
Yeah. So
Like,
we'll.
in between is kind of simple.
Yeah, I think they have two types of digestion. They kind of like digest on the outside, and then they also absorb and digest on the inside. This is my recent trying to learn about ctenophore waste products. So yeah, we might sample those. We will be trying to focus on the more delicate animals with the ROV for sampling, because we can't really get those with the nets. So it also kind of depends on what we see when we're coming up. But if I'm wrong about the ctenophore digestion, anyone can correct me. That's my understanding.
Hmm? I thought Doug said he did. So, no, I thought you considered it on deck to be 11.5 for it. Yeah. It's Doug. I think. Get all mixed up anyway.
Ooh, cool larvation house.
I mean, they should be linked to the drumming folder. Unless they've had the drumming folder off for no reason or loosened it, tightened it, something.
Hmm. Ooh, it's straight ahead. No, it's that it. Yeah, it's probably a particle. I'm kind of like on the lookout for the radiolarians, but I don't think that was one.
Yeah. Yeah. Take some of it out. We're gonna change your direction a little bit, Rich, just to counteract some of the current we're seeing.
Copy that.
It does kind of change that once we're getting we're at 1,000. It does change a little bit. Maybe it's just
Yeah, Robbie, Rich, I will adjust the heading slightly to report.
Oh, it'll be copies, thank you.
out system, so a 5,300 so and that's the one is 1,700 to 5,300 without. Yes. Wow.
What's that? Like bright red.
It's changed. So it said
Let's say
40 something, then it's at 5.
We were at around 5,000.
Yesterday we were 55.
Yeah, okay.
At different times and we're moving around. But that number just changed a little bit, so I don't know if you're flying that.
Yeah, that might be
I'm not sure, but 740 is on.
That was when he was showing just online, so it shouldn't be quite you know what I'm saying?
These round things, Colin, are those what you're thinking are?
I think that was maybe a little, I think I saw it, maybe that was a little like ctenophore or something like that. That wasn't a radiolarian. No. What are those? I think they're
Those?
the wormies. They weren't quite they weren't as transparent as the Keating
Yeah,
nets.
so maybe animals.
Yeah.
They never definitely got some of the nets. Yeah.
Maybe they're Keating
That one did
nets.
look like a
Yeah,
Keating.
because the head was
It's
it
just
had like
different
that
species,
shape.
maybe.
Yeah, maybe they're not as maybe as you get deeper they have to be as clear. What is that?
Or it could be a fire zone.
Oh, we can't stop. I can't stop.
I did get a really big fire zone in the net.
20 seconds left.
Oh, that's cruel.
We had 2 more depths too.
Yeah.
1,250.
Yeah.
Yeah.
4, 3, 2, 1.
Okay, Bridget, that's all stopped.
Copy that, coming closer.
Okay.
Go down.
So next depth is 1250. All right.
We could talk a little about radio. Do you ever see arm lifter, Colleen?
Oh yeah.
How do you tell the difference between them and the radio area?
Oh, well, at least the ones I've seen, the foraminifera kind of have this like
Wide out.
golden center, and then they've got like,
Depth.
you can see all the,
30 and scale. 60
it's like they're not really, they're like they're soft pseudopodia or something like that like come out. They look like, I mean, they're still small in the ROV, but they kind of like glow, but they're golden in the middle. And then the radiolarians that I'm thinking of, they're called phaidarians, and they're colonial protists, so they have silica. They look like a buckyball, actually, kind of like, and then they've got capsules, like 8 to 16 capsules of, that's the cells, and then they really reflect in the light, so they look like, I don't know, they're kind of distinct. So you can tell because they reflect on the ROV light a lot, and then if you zoom in on them, they're super beautiful. And they often have this trail, the other way is they often have a trail of marine snow detritus kind of like hanging off of them, because that's what they're eating. Yeah. I had a postdoc, Natalia, who is still studying them. But so that was fun. That was like a really fun project. We're still working on it, but so I was kind of curious if we would see any here. But they kind of blend in with the like larvation houses and aggregates, so you have to train your eye to spot them. Oh, that one. See that thing that was blurry right in front?
Yeah.
I think that might have been a rhizaria.
Rhizaria.
Yeah, like part of the radiolarian big group. Maybe. It kind of looked like
It's huge.
it was right up against the camera, so it wasn't that big.
But for a
For
single
a
cell.
protist.
Oh yeah, it's
Yeah.
huge for a protist. Right, yeah.
Yeah, I think it was hard to solve here.
They don't look that transparent compared to how they look when you pull them up in the.
That's one of those red ones.
We got into that. Yeah, there's a bunch of them. Yeah, they're really cool.
Like they look like it's a monocle, but then it's actually a table. That's what the whole is.
sitting here, it's not that big, it's more
This business is making a big part of this stuff.
yeah.
Maybe it's being overactive down this deep, but also small.
But not that small. And the filter ground, the deeper bottle filter goes with very brownish. Never
Really?
seen.
Stationary organs, none of these larger part of this on this step.
Oh, fish. Yeah.
Oh, nice.
It's far away.
Double shot.
That was pretty good.
Did you collect many fish in the mock nest? Were there many fish in there?
Some.
Yeah.
Definitely like a small, like just a fraction, but I saw it in there. But I also didn't necessarily see everything. They might have already.
Oh, yeah.
They got a couple of dragonfish.
Dragonfish?
Mm-hmm.
Which ones?
Today.
Are those the long ones? Or what? Yeah.
With the scary teeth.
Oh, cool.
Like they often have blurs.
We have to get to 1250. We just when we're doing the transects, so we have to stop and any bottles yet. We'll do like 10, 15 minutes at 1250, 15 minutes at 1500. And then we will start coming up, that's when we start taking samples. Yeah. Yes. So yeah, we would use the acoustics to figure that out. It's a good one.
Number 10. Number 10.
Meridian?
Yeah.
It's a worm.
Oh,
It's a type of roundworm. Or no, flatworm.
a flatworm?
Yeah.
All right.
They got some, I think, in the minutes.
Oh,
They're really cool.
maybe we'll see.
They're like
Have
they
we
have
seen-
these like patterns, which is their I think it's like their digestive tract, but it's really cool. I got your picture.
Have we seen any in the ROV yet?
Not
Oh,
that.
Oh, nice.
big long one.
Yeah.
Here. The ones that aren't spikes.
Oh, cool. Is that like a Tomopteris?
One of our scientists
that
from Argentina
portraits
just
or-
came
That's something
into the
different.
room.
Or annelids, but
Oh, okay.
yeah, that's like an internal structure that you're seeing.
Oh, I see.
Yeah.
They're blobby.
Yeah.
Oh, cool.
They can also get really big. I think we once got one and then Loch Ness was like literally this big.
Whoa.
Like
That's
bright
cool.
orange.
Okay, I want to see one of those too.
We have a scientist. Yeah, those are the ones that was
Yeah.
We have a scientist from Argentina in the room who's stopped in case anybody has questions to ask in Spanish.
Can
There's
they?
some already up there.
How good are the microphones? Can they hear back here?
They can hear, but it would be better if I activate that one.
Do you want to sit here and I can switch? Sure. Yeah, I think I
We're at 1250.
All right.
Or I can
12:50.
Start to move again, Bridge.
Moving.
Okay.
On the move.
You're going?
Yep.
Okay. I'm starting. Do you want to log it? Lee, you want to log it?
Sorry.
Okay. Oh no. No problem.
I just did it start?
Yeah.
Okay. This is 12:50.
This is 12:50.
Hola, ¿me escuchan? Mi nombre es Lucía, soy una investigadora argentina. Quería responder un poco a las preguntas que nos están poniendo en español. Sí, es la misma Sophie que ya conocen, es la misma persona de Schmidt Ocean que trabaja con nosotros para los streams. Hay varias personas que estaban preguntando por dónde estamos o si ya estamos en Brasil. Salimos hace exactamente 5 días de Puerto Madryn. Estamos en aguas internacionales. Exacto, como mencionan por acá en el chat, no estamos en aguas argentinas, estamos fuera de la plataforma, por fuera de las 200 millas marinas. Y ahora vamos a seguir viaje hacia el norte, hasta la zona económica de Brasil. Ahí tampoco vamos a tomar muestras y vamos a terminar en Río de Janeiro, pero recién el 22 de febrero. Así que todavía nos queda casi un mes que vamos a estar con este proyecto que se llama Bioreactors. La idea es poder entender un poco cómo los animales que viven en estas aguas de profundidades medias, entre 200 y 1000 metros de profundidad, cómo procesan la materia orgánica, o sea, los alimentos que consumen, los residuos que van dejando en el océano, porque todo eso contribuye a la captura de carbono. ¿Sí? Todas esas partículas que ustedes ven bajando en la pantalla son detritos, son partículas que están sedimentando, que están bajando a una velocidad muy lenta. Ustedes las ven rápido, pero la ven rápido porque el robot está yendo muy rápido, porque lo está arrastrando el barco, pero realmente bajan muy lento. Y esas partículas se van depositando en el fondo marino y así es como una de las formas en las que el océano captura carbono. Así que hay algunas personas, acá algunos investigadores que están interesados en esa parte del ciclo del carbono, entender cómo estas partículas se acumulan, y otros que están investigando la parte de animales, peces, cefalópodos, crustáceos, medusas, ¿sí? Todo lo que cada tanto se ven en la pantalla. No, para investigar en aguas internacionales no hay tantos protocolos. Hay acuerdos internacionales que limitan sobre todo la cantidad de muestras biológicas que se pueden extraer. Uno tiene que informar qué es lo que extrajo cuando termina el proyecto. Pero eso es un acuerdo bastante nuevo que, si no me equivoco, salió el año pasado. Lamentablemente no recuerdo el nombre, pero sí hay algunos acuerdos internacionales, pero no son tan estrictos como los que tienen los países, ¿no? O sea, nosotros, por ejemplo, no tomamos ninguna muestra ni ninguna medición en la plataforma continental argentina, tampoco lo vamos a hacer en la de Brasil. No es que no se pueda, sino que requiere otros permisos, otros protocolos que nosotros no teníamos. Entonces, en aguas internacionales es más fácil acceder a la toma de datos y a la toma de muestra. Ay, mi hermana está acá. My sister is here.
Oh, good timing.
Yes.
I want to see what is going. It seems like it's pushing over.
Claro, sí. Tardamos unos días en empezar porque tuvimos que navegar bastante lejos, fuera de la plataforma, y también queríamos acercarnos lo más posible hacia el este, porque bueno, si no saben, el océano no es uniforme, ¿sí? Hay distintas partes con distinta concentración de clorofila, ¿sí? Distinta concentración de fitoplancton, que son los organismos que, así como las plantas, fijan el carbono y producen oxígeno. Y en las regiones donde hay más clorofila, en general se encuentra más zooplancton, más peces. Entonces vamos pasando por distintas partes del océano, distintos pedacitos, ¿sí? Que tienen distinta composición. Entonces, para alcanzarlos a veces hay que viajar mucho, porque están muy separados, como esos biomas o bioregiones. Entonces, bueno, imagínense lo extenso que es el océano, lo largo que es el viaje. Y tenemos un barco muy chiquito, a pesar de que parezca tan grande, es muy pequeño y se tarda mucho en llegar a cada lugar. Y piensen que nosotros estamos tomando muestras de un solo punto. Hay infinitos lugares del océano que todavía no se conocen. Entonces, bueno, en el tiempo que tenemos, que es solo un mes, tratamos de optimizarlo lo más posible. Pero bueno, solo vamos a parar en 12 estaciones. Y es un montón de trabajo. Cada día en que paramos en una nueva estación, nos levantamos muy temprano. Yo hoy me levanté a las 2 de la mañana y otros compañeros también nos vamos turnando alrededor del día. Y nada, es mucho trabajo, porque realmente hay que aprovechar mucho el tiempo, es muy caro. Este tipo de barcos de investigación son muy costosos de mantener porque hay mucha gente que trabaja, muchos recursos. Entonces, bueno, es un proyecto muy ambicioso, así que tratamos de aprovecharlo al máximo. Claro, sí, es como observar una gota debajo del microscopio, solo que estamos observando a simple vista. Ustedes no están viendo ningún aumento en la pantalla. Esto es realmente lo que se ve en el agua, en el océano, ¿sí? Lo que pasa es que, bueno, la iluminación un poco ayuda a ver estas partículas que están cayendo. Pero sí, es un poco parecido.
Sinker. Or no, it's a siphanophore.
Oh no, that one is
I think so.
No, I think it's a
That big red thing?
yeah.
I think that was a siphanophore because it had the
Yeah.
bells and the scraping stuff.
Yeah. But the tentacles.
Yo estudié en la Universidad de Buenos Aires. Soy de Buenos Aires, de Argentina. Estudié química, no estudié biología marina y me fui tirando un poco con los años hacia la oceanografía. Y esas partículas que estamos viendo son lo que se llama nieve marina o detritos marinos. Son pedacitos de materia orgánica, usualmente de organismos que van muriendo. También ven algunas cositas que se mueven, pero en general lo que se ve son partículas que caen al fondo del océano. Exacto, materia orgánica, heces, sí. Como materia fecal de los peces, por ejemplo, de los artrópodos, del zooplancton, todos los residuos que dejan atrás los organismos van sedimentando a lo largo de la columna de agua. Y en el camino, mientras van cayendo, también sufren transformaciones, ¿sí? Hay bacterias que se alimentan de esas partículas que caen, hay otros animales que las comen, van sufriendo transformaciones químicas, ¿sí? Las moléculas cambian de forma y todo eso es lo que estamos tratando de entender un poco en este viaje. ¿Qué pasa con la materia orgánica cuando viaja a través de la columna de agua? Estamos en el Océano Atlántico, al sur, aproximadamente, no me quiero mentirles porque no me acuerdo exactamente la distancia, pero estamos a 5 días de viaje de Puerto Madryn, en dirección hacia el este. Las distancias se las debo porque hoy me levanté muy temprano y no me pude fijar. De hecho, estamos trabajando en... uno de los proyectos de este viaje es recolectar peces, artrópodos, todo tipo de animales de distintas profundidades. Y recién hoy a la mañana levantamos una de esas redes, que es como una red de pesca, pero un poquito más sofisticada, porque tiene distintas formas de medir la cantidad de agua, ¿no?, que pasa por esa red. Así podemos saber, tener un poco una idea de la concentración de animales en esas aguas. Entonces recibimos todo ese material y hay compañeros que lo están clasificando, otros que los vamos almacenando para otros estudios. Y hoy la idea es llegar a 1500 metros de profundidad, no sé si ya llegamos, y después vamos a empezar a subir de vuelta. Bueno, cómo llegué a ser parte de la tripulación fue mucha suerte y muchas oportunidades que se fueron dando. Yo vine a hacer el doctorado a Estados Unidos, hice mi doctorado en San Diego, en California, y de ahí me recibí el año pasado, me doctoré en septiembre, y muchas personas que hacen doctorados a continuación siguen trabajando en investigación y hacen algo que se llama un postdoctorado, que es una estancia corta de unos años en otro laboratorio donde uno sigue aprendiendo otras técnicas, sigue trabajando para publicar artículos científicos. Y en ese proyecto entré a un nuevo laboratorio, que es el de la jefa científica de este proyecto, que es la profesora Anitra Ingalls, y ya tenían planeado este crucero. Entonces me tocó venir, tuve mucha suerte. Yo no soy parte de la tripulación porque no trabajo permanentemente en el Falcor, sino que vine solo por un mes. Acá somos casi unos 30 científicos y otros 30 son personas que trabajan permanentemente en el Falcor. Y son fundamentales también para la misión científica. No, el ROV, el subasten no va a llegar a la profundidad máxima, sí, que es como 5.500 metros. No, porque nosotros nos vamos a quedar en las aguas medias que se llaman, que es entre 200 y 1.500. No vamos a llegar tan al fondo, por eso ustedes no van a ver el fondo marino. No vamos a llegar tan abajo.
What is that?
That's one of those things
Yes, we've seen them in the nets.
Oh, yeah.
Wow.
Siphonophore.
Oh,
that's
nice.
so nice.
Another one.
That was a good view. Oh, another
Yeah.
one.
Oh,
Wow. This is at 12:50. Okay. That's fun.
That was fun. People are very excited.
Yeah. Yeah, go for it.
what are they asking you?
They were really interested in what are the particles. So I spent a lot of time.
What are they?
Yeah, explaining like marine snow and
Really?
the tridos. Yeah.
They want to know what the particles are?
Yes.
Oh, that's like all I want to talk about.
Yeah.
I've been trying.
And then they are very kind of anxious of where are we, and I couldn't tell them like a good like analogy of like how far we are from other things.
We stayed we're 3 days away by boat.
Do you see the light bot commands in there?
Yeah.
Regularly, people will ask when other people know that there's a light bot command for the live link of where Falcor is. So usually that goes
So beautiful.
reiterate that. The chat's pretty iterative.
Okay.
Sometimes I like to come with
Somebody actually asked. I don't.
Yeah.
Somebody asked if Sophie was here.
I thought they know.
All right, that's 15 minutes at 12:50. So we can stop and then dive down to 1,500, and that's our deepest depth.
Okay. Heading down to 1,500.
Okay.
They were using station keeps and then keep drifting back like that.
You don't have station two.
Sure do. Station.
Ah, you're telling me to use. Ah, okay. Ah, I thought you said you are using station.
No, you're not.
I'm not using station.
Yeah, that's what they were using it, so they weren't drifting like that.
Thing is, I lose pressure. I was trying to kind of balance it out and
Well, you can do it if you want. You can just drift it backwards.
Okay, since I was given the prompt, if people are interested in the particles, this is Colleen. Yeah, this is Colleen.
How big are they? What are they?
Oh, I see. Our lab, that's pretty much what we study. We study the marine snow, the particles, and the biology of what forms those particles, what they are, how fast they sink, the animals that interact with them, because we're interested in how much carbon the ocean is taking up and how much sinks into the deep ocean. And that's because that's regulating the climate, because it's taking it away from the atmosphere, but also it's feeding all these animals. I mean, most of all these animals are living here and on the seafloor, most parts of the seafloor, because the source of their food is this sinking particles. And what's really cool is if you put these particles under a microscope or look at them really closely, they can be really beautiful, actually. And when you look at them, the shape and the color, you can start to infer what made them. So a lot of these particles are going to be fecal pellets, and fecal pellets from different animals have different shapes, different colors. And I mean, that's why I was talking earlier about, like, what does a ketognath poop look like? Because in our samples, we see lots of different shapes and colors of poop, and we are learning what animals those come from, so we can see which animals are controlling carbon export. And also, a lot of other organisms eat that poop, and so that's feeding a lot of the animals down here. The other cool things, there's a lot of stuff that just looks like detritus, like fluffy stuff, and that's can be formed by lots of things. It's degraded fecal material, but it's also things like the ctenophores and the jellyfish and the siphonophores. The way that they feed, stuff just kind of sloughs off of them because they're digesting some of their material externally, so it falls off, and that becomes marine snow that's sinking as well. Also, the larvation houses, those larvations that we've been seeing, those are a huge part of what's forming particles. And when those when the larvations abandon their houses, those houses start to sink, and they sweep up more and more particles as they're sinking, and they become these long, stringy sinkers that can also transport lots of carbon into the deep sea. All of those particles have a lot of microbes growing on them, different bacteria that are eating and breaking them down. And so the bigger and faster something sinks, the deeper it's going to get before all the microbes growing on them break it down. And if you're thinking about feeding deep sea animals or sequestering carbon, then the deeper it gets is, the more effective it is at transporting carbon into the deep sea. So that's why we are look at all the details of what all these particles are, because we want to kind of find clues about what's controlling how much carbon, the biology of what's controlling how much carbon is being exported. So I could just talk this whole ROV dive about poop if that's really what people wanted. Oh, snot house, yeah, or a snot palace, as my colleague Kakani Adambari likes to call them. Yeah, so the snot house or the snot palace is made by the larvations. Also, yeah, that's their house. Yeah, there's a lot of my colleagues love the snot palace.
I see a lot of little things swimming around, maybe zooplankton at this depth. Is that, compared to just above this?
Yeah.
Do you agree?
Yeah, it does look like there's more stuff swimming, right?
Yeah, little, I don't know.
Yeah, just like more things zipping by.
I feel like we've seen that for like the last 500 minutes.
Have we?
Maybe not.
Okay.
Oh, it's
Maybe
kind of
the
boring.
company
This is
won't
why
have
we
it.
got a recording. We can double check.
This is why we do
Yeah.
quantitative transects.
Yeah.
So we're not going on fives.
Yeah, no, but it does.
Right.
I feel like I'm noticing it more here.
Does anything eat larvation houses? I mean, there's a ton of microbes on there, and both bacteria and protists. There's tons of heterotrophic protists also. Those single-celled things for sure are eating them. You know, I bet for sure. I bet there's tons of like these detritivore copepods that just actually they specialize in attaching to detritus that's sinking, and they're eating the larvation houses that they kind of look like marine snow or detritus, but it's an abandoned house. Larvations are weird. Oh, yeah, I guess so, but I guess if you think too much about any animal, you could kind of say that. Maybe humans are weird.
I mean, for me, it's really interesting because I generally study particles on a filter that have been filtered, and they just look like some gray, haze on a filter, and just being able to see what they actually look like before they've been put onto a filter is really amazing.
Yeah. You can just like appreciate the complexity of the biology a lot better.
Mm-hmm.
Oh, today's dive is going really well. It's our first dive of this cruise. And there's a lot of people on this science party who have never used an ROV before, so it's pretty exciting for a lot of people to like see this kind of view of what we study for the first time, and I think it's going really well. We're gonna try to do like 2 or 3 of these at every major station that we stop at. So
You need to tell them that you are not inside the ROV.
oh yeah,
People
I will, yeah,
something
just
that people
yeah, we are
usually
not
ask.
inside the ROV. We are sitting on the ship looking at computer monitors. The ROV is like a robot hanging off the ship with a lot of amazing, like 2 extremely beautiful cameras, just super high definition cameras, and we are watching that, and I'm sitting next to extremely skilled ROV pilots who can control that robot remotely. So yeah, just in case you thought we were in underwater, we are on top of the water. We're almost to 1500.
Yeah. Going to go.
We are technically not in Argentine waters. We are out in international waters, but due east. Got my direction correct this time. Due east of Argentina. Yeah, Puerto Madryn is where we left from, and we just basically went east.
We sailed for 3 days.
Yeah.
At 10 knots.
Mhm. But we're very far offshore.
Your 3-day steam offshore going 10 knots.
I'll try. Oh, they got trim dialed in. Touch that button.
No.
Yeah, there you go. That's why you had trim it.
I pressed.
Somebody had trim it.
Mhm.
Just telling you, that's why it kept going down. Now you can go it kept not going.
All right. Okay, so we're at our deepest depth, 1500. Ooh. Oh, that's cool. Was that a
It was a
Tina 4? Yeah, it looked like. I saw the
You're drifting back quite a bit.
It's so interesting. I think of the deep particles that we measure things on as being predominantly not living, but there are so many living things swimming around that we can just even see with this camera. It's incredible.
Yeah, I mean, the chances of catching them in a Niskin bottle is like very low, though.
I think about how many Niskin bottles we do. Like,
Oh, look at these
kinda crazy
particles.
differences.
Okay, are we driving forward now?
Not yet.
Oh, not yet. We're still
Yeah.
getting
I
to the right
overshoot
depth.
down, so
Yeah,
we're coming
you're
back
too
up.
excited.
I mean, I guess I've seen like, because I want to go tentacles directly from the top.
Okay.
We're getting at big animals, but the monkeys are very small.
All right.
Well, there's also
Okay,
like, is the
we
SOI
are ready to start.
I'm starting to get some
Just let me
momentum
know.
forward.
How? Tell me when you think you're.
I'll let
You can
know.
just move. You can just go, and then we'll start the timer.
How do you think this data can be used to get particles? Like
I
carbon
thought about
concentrations?
that. Yeah, it's. I think there's. I've been brainstorming for years with Chrissy, who works with me, about because like just so many years of ROV dives. It'd be so cool if we could get just particles, right? Just
Yeah,
like,
carbon.
but it's tough because of the sizing is tough.
Mhm.
Okay. I'm
Okay, we're
good.
going. So okay, I'm gonna start the timer. But one idea we had was, yep, was like,
They're really excited. That's okay. They'll ask you questions.
yeah, they'll ask you, just introduce yourself.
We have another scientist in the room from Argentina.
Yeah, just introduce yourself,
We're
and
gonna put you on the
they'll ask
spot.
you questions. Because some people don't understand what we're saying, and they don't know what's going on. Yeah, yeah, yeah.
You can
Or
also
you
just
just
answer
go-
questions.
They're coming up on the screen with questions, and you can read them. And just
And I think that microphone's on, so
I can turn it back on. It's not.
and it's also the person's birthday.
oh yeah.
Happy birthday.
Thank you. Okay. Oh wow, this is a cool depth.
Yeah, I just turned your microphone on so we can chat.
Bueno, hola a todos, me estoy sumando recién. Soy de Argentina, soy bióloga, me llamo María Luz y bueno, estoy acá viendo y tratando de
Is the current stopping you?
traducir algunas cosas.
It's going too fast. I'm trying to reduce the
Okay,
time.
maybe we need to restart it.
Ay, gracias, sí, también es hoy es mi cumpleaños.
I don't know. What do you think? What's
1500 seats up in the top left. Yeah, so this is our last video concept, and then
pause
we're
it.
gonna
Let's
head up. But head up slowly and like
Yeah,
look
let's
at
start
stuff
it.
maybe.
Ready to go?
Bueno, el máximo de la profundidad de hoy va a ser 1500.
All right, we're starting.
Ahora llegaron al máximo y van a ir subiendo. Bueno, gracias por los saludos.
Are we starting now?
Yeah. Okay. Oh.
Vamos a quedar grabando ahora los organismos que están a media agua. Ay, muchas gracias por los saludos. Sí, así es.
Use this that's this
Notice.
It's still pretty early on the West Coast.
No, no iremos hasta el fondo hoy. El objetivo de esta campaña es ver los organismos que encontramos a media agua y que también estuvimos durante la noche pescando con una red especial.
Do you know what the
A
speed
little
is?
over half an hour
Okay. So it's been the same the whole time, basically.
for the most part.
Yeah.
So probably,
Yeah.
before we start ascending, I would like to fire Niskin bottle like 1500 meters. We will probably get 3 bottles like in the amber's area and 3 here in the area.
Oh, that's
Oh,
a
that's a-
larvation house.
Yeah.
So I don't know if you want to use the EPA to determine where the amber's area is. You know? What is that?
You tell me what depths.
This is live. This is on.
Bridge RV updated waypoint.
Okay.
Alex is not in the control room, but he is on the ship. And he's taking really beautiful images, like some of the most beautiful photos I've ever seen. He's so good.
Yo estudié biología en la Universidad de La Plata y después me mudé a Mar del Plata a hacer mi doctorado y ahora estoy trabajando en el INIDEP.
Is the current pushing us back?
Y
This stuff seems hard. Harder than the ice.
bueno, llegué al Falcor.
Stop.
el año pasado me surgió la posibilidad de hacer unos estudios abordados en esta campaña. Y bueno, acá estoy.
Since up there, we're not accurate, it's about 5,000 feet.
Creo que la profundidad de este lugar es como 5.000 metros de profundidad. Y solo estamos, el ROV está a 1.500, ¿no? Sí, lo puedo ver en algún lado. Sí, ahí está. Gracias, Karen.
Beautiful.
What was that?
That was a larvation. I think. I'm pretty sure because they have these like two
Yeah,
lobes, kind of. I don't know if that's the right word, but
I think it is the bone lobes.
okay.
I love the shrimp with the soup and all.
Feels like there's a lot down here, right? Or no?
A lot of work.
Animals. No? More than some of the other?
You mean the other depths?
Some of, yeah, doesn't it seem like there's still a lot of
I think it's
animals?
we're also
or-
biased by like the animals down here are less mobile.
Oh,
So
yeah.
There's probably more animals up, but
They're
seeing
swimming
them.
away from the ROV.
That'd be my guess. Yeah, like more larger fishes and squids.
Mm-hmm.
Well, like the high pitch one
No, and all these
Yeah.
kids.
I guess the ROV sounds really quiet to us from here.
Yeah.
Not down there.
1,500
Yeah.
years away.
Not down there.
Yeah.
So many cuticles.
Is study data going to use only the science cam footage? I think we'll probably only use the science cam footage because it's kind of going straight forward. And that's kind of typical. I guess we could try to use the other one, but we kind of set the camera up in a controlled way, the science cam, so it's standardized for every depth. Yes. Stealth mode. Yeah, I'm not sure. Maybe we just need like silent scuba divers that can go that deep.
Well, that's like the good thing about things like weighted camera systems.
Right.
Like a noise.
Yeah.
That's how you see the bigger
Yeah. Yeah, if you can just like leave a camera quietly in the ocean for a while.
We'll usually have the lights off except for like turn them on periodically to see what's there, but then it's like not as intrusive.
Mm-hmm. Let's see, what do we got? We got 5 minutes left on this transect. See this? I think, what's that? Oh no, that's some kind of a gelatinous thing, huh? Someone asked what percentage of the critters will migrate up and down. Oh, I think that's an Anella question. I don't know. We got to get Anella. She's in the lab. She's either hopefully taking a nap. She's probably in the lab sorting animals. So.
She's there.
Oh, she's here? Oh no, Anella. I know. Their names are so similar. Yeah. Anella's our on-the-ship expert on animal taxonomy and their ecology, ecology and behavior. We're all learning from her. We did have a krill layer.
That's true. And up here, the surface is a lot of saps.
Oh, and we had a Ketignath layer where it was just tons of Ketignaths.
Yeah, but they're also
Oh,
right
let's
there.
look at that particle. That's a cute, yeah. Hmm.
Did you see them?
Okay. That's 15 minutes.
Grid, you can slow a stop, please.
So now Anitra wanted to fire. She wanted to fire one of the Niskin bottles.
Don't fire a bottle.
No. She does not want a bottle. No.
Changed our mind.
No.
This is
Okay.
what happens
No
when
bottles.
when we see the data, this
Yeah,
is
yeah.
what happens.
Okay.
Change our mind.
All right.
Um,
So what?
so I
Do you
don't
want any bottles?
Not yes, but not here. One of bottle at $8.75 will be the first one.
How many?
Yeah.
You want all of them?
Yeah.
How many
no,
are there?
2 bottles at $8.75, 2 bottles at 500, and 2 bottles at $3.75.
Okay. Wait, give me those steps again.
Okay. $8.75.
8.75. Yeah.
And 500
500
and $3.75.
and 3.75. Okay.
Those are 2 layers.
We'll make sure we hit those.
There's 2 layers on the EK80 that show a lot of animals, and 1 layer that doesn't show very many.
Okay. So I think, well, the plan is now is to kind of like go into like slow search mode as we slowly ascend. I mean, we have plenty of time as far as, the way I calculated the time was, assuming if we were down at 1500 and didn't do anything except need to
Come up,
recover,
yeah.
it would be an hour and a half.
Correct.
So that would mean we would need to leave at 12:30, start ascending from 1500. So that means we kind of have almost 2 hours of like playtime.
Okay. And what is your goal? What's your tack? Are you trying to stay in this position and come up, or are you wanting to move forward and come up? How are you wanting to do that?
Whatever's, I think we want to slowly drive forward to search for things,
Okay.
right?
All right.
And I think in terms of the direction, maybe whatever is, whatever would be the direction that's going to make it the easiest to collect. In terms of the current flow versus the ROV. So I think we don't really care now which way the flow is. Whatever makes it easier for you.
Okay.
And then the other thing is like we're kind of the lighting is really good for spotting or like quantifying stuff right now, but we might wanna make the lighting beautiful if we want to do like
What's the red thing?
a zoom in on something.
Yeah.
So just saying that out loud so I don't forget. But maybe we could do a slow search at this depth for a little bit and then start to ascend. So now there are no restrictions on zooming or changing the light or anything. If we want to like look at this jelly a little bit, then let's do that.
There's also
That's the water on for rinsing.
Oh, okay.
there's also a yellow ctenophore to the right. To the
Wow.
right.
Okay, that's really beautiful. Do we want to prioritize things that we feel that we can get a gut sample from?
Yes.
So
Yes.
I will defer to you.
Yeah, she said I can change them.
Or if there's anything you're like, I really want that for the gut microbiome, then
And things that we haven't seen so much in the nets, right?
Yeah,
that would be
they got a few of
good.
these
The
guys.
things that are more delicate that the nets really can't get.
Easy 50s.
Yeah, they were a little messed up looking, but
It's okay. Yeah, so not get this one maybe then if we see them in the
they
nets.
also,
Okay.
I don't know, you mean gut microbiome would be more tricky from like
Yeah, this one I feel would be a little challenging.
Yeah, that's fun. It's gorgeous.
But also we should maybe target the most abundant things too.
Yeah, we do.
Like
yeah.
the things that when we do the quantification with the video analysis, we see are really abundant and make sure we
I
have
don't
those.
see share.
Yeah, it'll come back. Yeah, too dark for me.
What's our, no, but what's our hours? Yeah, if you open up the hours.
Is there there's no scale bar anywhere in real time, is there?
No, I mean if
If it's big
If-
enough, you can get the lasers on it.
yeah, I don't think the
Hey?
I'm
It
curious.
has to be at
Less
least
than
10
10
centimeters.
seconds.
that thing's I'm guessing that
You're
thing's
playing with the lights.
That's like a couple centimeters. Yeah.
The ones
It
that
looks
they caught
huge
the
on
net
there,
are, like,
but
I
it's
know, that's why I
not
wanted,
very
my
big.
scale bar is not
Yeah.
calibrated in my mind.
Yeah.
Or these make things look so much
Like,
bigger.
yeah, sure.
Because their cameras are just too good.
Yeah.
Um,
But
I
it's all the more incredible to see like blown up with large
wow.
shadows.
I don't think we need to collect this one. Yeah. Okay. I'm over it. Let's move on.
One more step.
Leo,
Yeah, let's.
stick around for about 10 seconds.
Okay.
They're trying to get some video.
Yeah. Yeah. That marine snow is pretty beautiful too. Perfectly in focus. Yeah.
They're gonna want to get Nancy there.
It focuses.
Oh, you're drifting way out.
I'm losing my reference.
Yeah.
Can we continue surveying? Or looking for other things? Whoop! It's going away.
You start going
We're
forward.
gonna organize these tentacles.
Without even having a brain.
I mean, sometimes they're just all over the place and then suddenly they go into action.
Do you have an idea of the size?
She thinks a couple centimeters.
Okay.
Start making your way forward.
Big proximity and that's.
Should we? What would you prefer to search fast like this or slow?
Me?
Yeah, I mean, just so if we see something, we might like. Do you think we'll have time to like stop quickly to.
It's because I'm drifted far
I
away,
can
so I
move
need to
over
go
there.
Oh, I
back.
see. You're repositioning.
Yeah.
Okay.
We're just staying depth
Great.
right now.
Mm-hmm.
Well, we can move the vessel.
Yeah, we can move the vessel if we need to.
I'll slow down, so if you see something, have time to wait.
Okay.
The ship is the RV right now. How far away from the ship is the ROV right now?
Yeah.
80 meters at least.
Wow. Okay, I think we'll pop the other way. Almost. Amazing. I know.
120.
Football, soccer field is what I meant. Most of the world calls it football.
It's true.
It's almost a Super Bowl in Seattle playing, so.
Oh, really?
Mm-hmm.
I remember when I won the second year.
Yep.
That because of this.
So Colleen, how long do you want to search at each depth, or are you just gonna call it as we see it?
Yeah, let's just call it.
Okay.
And it's fine if we are like, it doesn't have to like stay exactly at that depth. It
Okay.
can,
Yeah, because I came up like 20
yeah,
meters.
that's fine. You don't have to go back down.
Okay.
Yeah.
I wouldn't do that.
The idea was just to use, we have quite a few hours left, so we can just slowly search for things of interest, either just to film them and get good looks at them, or if we think we can get good biological samples from them, then we'll collect them.
You guys not gonna do some suction during this?
Yeah, it would be either the D samplers or the suction sample. I guess it depends on whatever you think is going to be the easiest for whatever it is.
Because of the suction sample, we can leave it running.
Okay.
Sucking. I don't know if it's worth for you. Does it see
Um,
there? It's there in position.
Yeah.
We leave it sucking. It's gonna suck all the
Oh,
time.
Yeah.
I don't know if it's
Yeah, I think.
something that matters for you guys or not.
Is it if we want to use the suction sampler? Can we turn it on when?
Yeah.
Yeah, I think that would be better.
What time were the animals descending back? Did that start at like 3 or 4 or 5? I remember it being early for what they were expecting.
Do we want to try to collect libations?
I mean, I would like that. And I think there's an argument
Yeah, I think they'll show up in our sediment traps quite a bit. Anella didn't really care what we collected, so she was fine with us just collecting whatever we wanted.
I think if we're gonna collect them, we should probably go really slow because you don't see them until you're pretty close to them.
Yeah.
Usually.
And do you care
They're
what
so
depth?
delicate.
Do you care which depth you would get one from?
I think it would be neat to get them from multiple depths
Yeah.
as a comparison of like changes with depth.
And also maybe collecting discarded houses if we see them.
Yeah. So I think we could, since no one else, unless somebody else speaks up and says, or we see something really cool where we're like, oh yeah, we really need to get the gut of that.
Yeah.
Otherwise, if we see a easy to collect larvation house or a larvation, maybe we'll try to collect those as we
You
ascend.
need to tell me
Okay.
what is a larvae.
Yeah,
Section
I'll
or the
show
jelly-
you.
The section
Yeah.
or the jelly jar?
Yeah,
The section
get
with this
shredded.
corner part, right?
Yeah.
So, and then, so we could do something else with the section, doesn't matter if that's.
Megan wants us to try to chase down fish.
Oh, I think that's gonna be hard.
Yeah, we're not gonna get the fish. I mean, we're not getting this one, but like that's one right there. They're they're really delicate, so we'll have to kind of slowly sneak up on. Well, I mean, they're not gonna swim away, but I mean like so that the turbulence doesn't destroy them. But we can try. We're also interested in the ones that have been discarded, that just look like sinking balloons.
I mean, they're the things that I keep seeing kind of just disintegrate, right?
Yeah,
Like,
yeah,
almost
yeah.
exploding when they see us.
Yes.
That's not what we see.
I know they're not seeing us. There's some kind of current custom. There, like that one, just kind of.
Yeah,
disintegrated.
yeah. Yeah, like
Off to the right.
that. Can we see what that is on the right?
Oh, I think it's a siphonophore.
Is it?
Yeah.
Oh,
Maybe a tuniform.
no. Can we zoom in on that?
Yeah, I'm closer.
Oh, it looks like a tuniform. Oh, maybe. Oh, yeah.
Like broke off.
I see. What? Yeah, like it's a. Yeah, right. That looks like a siphonophore.
Yeah.
Cool. I guess it's probably. Damn it! Is that? Oh, see, look straight ahead now. See that?
Mm-hmm.
Oh, yeah.
Like, can we see what that is?
Closer.
I think that might be a larvation house. No, what is that? What is it?
What's going on there? 'Cause it looks like
Whoa.
a larvation, but then there's something on there.
Yeah, can we zoom on it?
Coming closer.
Okay.
I like this thing's
Whoa.
better than I mean,
Whoa, what
whatever
is
it is,
that?
I kind of want it.
Doesn't it look like... I wonder if it's like a... This is like complete speculation, but I wonder if it's like... Oh, kind of like an egg... No, like
What are you saying?
like eggs or like what do you think that thing is? It kind of looks like a
digestive tract.
yeah,
Like,
right?
It looks like an animal, but it's like.
I see the digestive tract,
Is it like an animal that's
like an egg mass coming out, or am I just? I don't know what. I'm not. I don't know
I don't know what that is.
that much about all these animals. So. All right. Well, I think we do not want to collect that.
You don't want or you want?
Oh, look, somebody on the chat. See, the people on the chat probably know the taxonomy way better than us. Somebody said it's a galaxy jellyfish.
Leave the autofocus on.
I don't know if they're referring to what we're looking at right now, but
Yeah, but I don't know. Who's controlling the focus?
We're trying because the autofocus keeps going away.
Yeah, I
So we're
see
manually
it's
focusing.
connected. That's what I figured.
all right. Well, I think we're not collecting that. And since we have 1,500 meters to survey as we ascend, we should move on. Maybe I have a short attention span.
No,
No, I'm trying to prioritize because if we do want to collect something, it might take us a little bit of time.
We have limited time, so.
Okay.
Okay, keep going.
Yeah.
We'll zoom out.
And I think we could slowly ascend actually, just like if very slowly, like if we're going any direction, it should be up.
We don't seem to be moving forward anymore, so I'm just gonna send it back. Reg ROV, got an updated waypoint.
So wait.
What you'll find, Colleen, is what's happening.
Mm-hmm.
Obviously, the animals are moving with the current.
Mm-hmm.
So if we try and fight it, they'll pass by us.
Okay.
So we're drifting back with the current to
Uh-huh.
try and keep these shots and everything. So
Yeah.
I'm just going to continually just move the vessel aft as well.
Okay.
That's no problem. I just want to let you know what we're doing.
Okay. Yeah, that makes sense.
So he can kind of move forward to see animals, and then once we catch him, he's kind of drifting back with it.
Yeah, so it'll
You
they'll
understand?
it'll be
We don't fall
easier.
behind.
Right,
too
yeah, it'll
much.
be easier, so
Yeah.
they'll just whip by the camera.
Okay. That makes sense. All right. Yeah, I was curious about that too.
Mm-hmm.
Big, yeah. To try, that could be a sinker, but yeah, that actually looks a lot like. How big do you think that is? Is that really close to the camera?
Not really close, no.
Yeah, I think that was a sinker. So that was like a. I think that was a. Like an old abandoned larvation house. Because it was so.
I just straightened it.
Yeah, I mean it could easily have been.
You see
Yeah.
from the camera above, you can see it's crooked.
Oh yeah,
something
what's
here.
what is that?
Is that
It looks like
What is it? Is that like an old siphonophore or tentacle or something?
kind of looks like it, but huh.
Did you say it's
Can we zoom on it or?
Flying, I'll zoom for you.
Oh, what's that?
It's all gonna break up. But
Oh, okay. Yeah. It's I don't know what it was.
sort of weird jelly nut? Maybe it's like
Yeah, it kind of looked like a siphonophore, but it was
also like an old, very old one.
dead or something. Yeah, it did not look alive. What's this guy? Is this something that they were able to collect in the nets, or?
They got some ctenophores, but I think they mostly got like the rowing, like the more robust ones.
Mm-hmm. Wow.
That guy's really
Wow.
delicate.
It's so beautiful.
I guess, yeah, do we want intact ctenophores?
I can zoom like flying.
Okay.
Do you see this? Do you think that's the digestive part, the
Yeah,
orange?
the red in the middle.
Yeah.
That's probably what it's eaten. Because I just did a little quick search on their digestive systems.
Uh-huh.
They have like a cilia mill.
Shall we try to collect it? What do you think, Anitra?
Oh, it's
Are you
got little
interested?
green fluorescing things. Wow.
Just iridescence.
Yeah.
Like, how much time would you want to spend trying?
Capture it.
Let's try.
Should give it a try, see how it
The
goes.
jelly jar? or
Yeah.
something?
I don't know how
What do you think?
fragile they are.
That pen is really fragile. I think you need the jelly jar.
Okay.
Are you gonna dissect it? Who's dissecting it?
I mean, my team is going to come and get the CTD water. They can also come and get that and dissect it.
I can't dissect it until we can pour the trap.
Yeah.
I could do it after.
Or Delano. Delano's the other point person.
I'll adjust the camera down for you.
Mm-hmm.
I was charging.
Are you ready to log this?
Me
It looks
neither.
like they're getting ready to collect it.
I'm just gonna log in the video though.
Mhm. Yeah. Good point.
So you're right. You see it?
Uh-huh.
In that camera?
Yeah.
If you need me to, I can lean over and run the balance too. I'm just gonna lean over you so I can get on the correct
Mhm.
wall path, okay? You tell me where you're,
On the bridge?
it's on top of it.
I just
Go ahead.
Can I just click the white and you want me to continue moving a step?
Yeah, please continue, thank you. I'll move the waypoint.
It's hard to
Yes, continue moving a step.
That's updated there, Rafael.
Copy, thank you.
Number 5.
Okay.
Trying to find some.
Is it in there?
Uh-huh.
Okay.
Yeah, I was moving.
It was?
Yeah. I don't know
Okay.
if it's still
It's probably not.
We can went through.
Mm-hmm.
Come on, let me see. Sorry, I didn't see it.
Oh, there.
Fast enough.
That would be nice.
You see it?
Mm-hmm.
Down
All the
below.
way down there, okay. There it is to the right. Okay. Passed it by.
Solid.
past the camera.
Yeah, it's fine if we lost it. If that's.
It's about quite a ways up. Yeah, it's probably gone by now. Probably caught in the corner.
It escaped to live one more day.
You need to put a little more weight because it's too light, and you need to correct, and it makes it more difficult to line up.
But we can just keep trying.
Yeah.
Right down there is where we're going to football fields up front. I want to solve it.
Over there. Yeah, removing one. In there.
Do you want to keep
See
going up
this?
one?
Yeah, I think if we're going in any direction, it should be, I mean, up or down, it should be up. So
Yeah.
can we, maybe we can check out that one? Maybe there's try number 2.
Mm-hmm.
There's one there and there's one there to the left. There's two of them.
Yeah,
There's
we
one
should
off
try to
screen
get
and
one.
there.
They're clearly abundant.
Yeah.
We think it's another unicorn, the same kind. It looks like the low, but it was a joke.
It's moving too fast.
That one's lost. Oh, there they popped up on screen. See it? It's to the right.
To the right.
It's going by us again. Updated waypoint bridge.
Copy.
what that actually translates to in terms of the size, but I know it's gonna be different sizes. So like that could be
You want to
krill
continue on, Colin?
versus
Yes.
squid.
Yeah. Can we slowly maybe let's see, we're at 1450 now. Let's come up to like, I don't know, 1300, just to like
All right.
slowly be coming up over time.
Mm-hmm.
I wonder if we could, like, would this work if we drove slightly forward and up and then tilted the science camera upward? Would that be a search mode? Would that be a good search mode? I guess the lights aren't, that it's kind of dark up there, huh?
Yeah.
Okay. Right, because when the ROV is coming up, it's still like, it's not the whole ROV is tilted upward. It's actually like still horizontal.
So that's kind of,
Ah, okay. So then it doesn't really matter.
right about there. It's kind of even.
Mm-hmm.
Straight out.
What depth you said?
I think I said 1300.
You know, I'm looking at, I'm redoing my water samples and depths.
Uh-huh.
So I'm looking at these.
Yeah.
Carefully. And I wonder if it would be, that we could try to capture things that are in the depth layers where I think I want to try.
Sure. That's a good argument. Let's do that.
So 825, I think, is about this layer right here.
Okay.
I think that says 800. It's very hard
I
to
think you
read.
can put the mouse over it and it'll pop up with the depth.
I don't have a mouse.
I got you. Here we go.
Okay.
Bridge updated waypoint.
825, something like that.
Okay.
30.
So what if we just kind of come up relatively rapidly to 830?
Yeah.
And then we'll just spend a bit of time searching there
Yeah. Yeah.
Okay.
And then
until we get
the
something.
fire bottles
Oh,
are
nice.
there that doesn't have any animals as a comparison.
Yeah.
So we don't have to linger there, but I would fire the bottle there.
Unless
Okay.
it's something like this.
Yeah.
And
over
then
there.
I would maybe want to try to collect something in this.
Something here.
Oh. Yeah, let's check that out.
If we see something cool, we can collect it down here. That's also.
Okay.
Yeah.
Okay.
Oh, it's a snipe eel.
Wow.
They've got these really cool teeth that are like serrated, like Velcro, and they use them to entangle shrimp antennae.
Oh, cool.
Wow.
They also stick to nets really well.
Oh, wow, cool.
Wow, look at that mouth.
Yeah, if if you feel them, it literally feels like Velcro. They're deep. So
Can zoom in so much.
saving energy.
And then there's something vertical behind it.
Yeah, it looks like
Wow.
a shrimp or something next to it.
I mean, yeah, is that an arrow worm? I think that's an arrow worm
I
that's
thought it
like
had
right
two
next to it.
eyes. Oh no, you're right. It just was. What if it
Um,
eats it? Just goes right in the middle right now.
Is that a fish gut that
We're not gonna be able to catch
no,
it.
it'll swim too fast.
Yeah.
Yeah. But we
It's
can
still
look at
now,
it.
But we're gonna scare it.
Yeah.
Really soon, and then it'll.
Mm-hmm.
Really cool.
Wow, this is incredible.
Put on the jelly.
So Lee, who's a postdoc at Mbari, I'm answering people on the chat. If you, I can answer you in English. Lee thinks that's called a snipe eel. Oh, another fish.
That looked like an echidna.
They all just kind of
Go for it now. I'll have the other one as a reference.
Look at those. Are those photophores on the side of it?
That's,
Look at those dots.
It might be, yeah.
Oh my
It's
god.
either photophores or just the lateral lines. Wow. Actually, yeah, because look at it on the mouth as well.
Oh, whoa. I wonder what it looks like if it's bioluminescent. Like, that would be so beautiful. Oh my gosh.
It's almost inside the ROV.
Woo!
That was quick.
Yeah.
He did not cooperate with that, Alex.
That's when we don't try to
Cool.
Just something leaving. I've marked it.
That was cool.
Snipe eel. That's what we think the name
Yeah, I'm
is.
pretty sure.
Yeah.
Okay.
Okay. So keep going to 8:30. That yeah, we said 8:30.
8:30. Yeah.
Updated waypoint bridge.
Copy.
There are a lot of I don't know if those are all the same species, but there's a lot of those ctenophores. What's that?
Yeah.
This-
I saw that too.
Oh yeah. The engineers
Ooh, look
that
at
I
that.
work
On the
with
left
love
is a
Solnesis
great
because it doesn't move.
The lower camera on the left.
Oh yeah.
It's a
The-
Tomopterid.
Mhm.
Which one?
It's like the one on the left, the thing with all the.
Go up a bit. Straight ahead.
Oh yes, right there.
Ooh, there's something behind there too. You see that coming in the distance? It's so beautiful.
super beautiful.
Yeah, go for it.
Ooh. Wow.
Yeah. Wow.
I see a gut.
I see a gut too. They're pretty fast though.
Yeah.
Pretty quick. It's
Section
all gut.
sampler.
It's all gut. I've seen an attempt.
Yeah, it's that's a. It looks like an 8. It looks like an 8.
Looks like it's got like one amplify in there.
Oh yeah, look, we can actually see what it ate. Oh my gosh, it probably, yeah, I wonder if that's just a little amphipod.
That's what it looks like.
Yeah. What's inside of there?
Like glass.
I mean, I'm looking at the shape of that thing right there. Wow. Oh yeah, it's so crazy. You can see its whole digestive tract.
It's like it's all digestion.
Yeah.
All you need is
Wow. Oh my gosh.
You see that it's not swimming.
Wow, look. The camera is so beautiful.
It's incredible. You guys make it look easy, but I know it's so hard to stay the same distance from me.
Do you know what they eat? Are they
You can see it in there.
moving.
Oh no, is that in the gut?
Digestive tract.
Whoa.
Is it in the gut or was it on the outside? Is it inside?
It's inside.
Is it? But we're making it puke. It's going the wrong
The
way.
amount of touch.
I know. I thought so too.
Yeah.
Yes,
Oh
sir.
no. We made it throw up.
No, it's still in there.
Oh my gosh.
It's going for more.
What?
Yeah, I think that the predators.
No, look it. Oh no, we.
I can zoom.
Look, it's.
Go for it.
Okay, zoom.
Oh gosh. Oh no.
It's coming out.
No.
Yeah, we made it throw up.
Oh no. He scared it to a I know he's trying to chase it like, don't, you can't leave.
Is that its butt?
You gotta stay.
No, that's his head.
That's the problem. It's the run
Only a mouth and it goes in and out the same
Yeah.
way.
Pull on the jelly shot.
It's a, no.
Updated waypoint bridge.
We think it's throwing
Got
up.
it.
Right, but did it like partially digest and then now it's still
Well, it was pretty far down the track.
I know.
That's their poop.
Okay, one more zoom.
It was definitely inside the gut.
It was for sure.
Yeah. Oh, I'm sorry, buddy.
Oh,
Oh.
yeah. Spooked him. Alright.
Okay,
You
well,
done torturing him?
yeah, you no longer have anything in your gut, so I'm not interested in you. Oh, we made him throw up.
Let's come back.
There's something behind it.
Oh yeah.
See it to
Do you
the
want to
right
check
there?
that out?
There on the back. On the back there.
The
Brownish.
yeah, this let's see what that is. It's probably a siphonophore.
Yeah.
Yeah.
Ooh, siphon. That's cool. Wait, I can't do this.
Oh my god.
Look at its like
Whoa.
sea of stuff around it.
Yeah, that's perfect lighting.
I'm almost touching it.
Okay, no worries. Let me know when it's cool to zoom.
I'm touching it. You see that?
Yeah, it's got it's like almost in the lens.
Yeah, it's like
He's gonna zoom a bit. That's why the lighting is a bit weird though, because it's in the
Yeah, I'm the shadow.
Yeah.
Almost into the jelly jar. That would be a cool one to catch.
Alright, I'm gonna bias the
do you want to get how do you dissect the gut of a siphonophore? I don't yeah.
That's a siphono no more.
I-
Siphono.
Oh, no more.
Gone.
Yeah.
Yeah.
Okay, see, look.
They're very
But-
sensitive.
Okay. Well, let's still make
Keep
our way
coming
up
up.
to
Yeah.
8:30.
We stopping every 2
Yeah, I know. We're
years.
just seeing too many cool
This is
things.
typical.
It's typical, we could be here for the next 48 hours.
Yeah, it's
Yeah.
like once you see something, there's so much cool stuff. There's just not enough time.
Every little corner
Yeah.
and all these particles is so
Yeah.
cool.
It's,
Ooh,
And your camera is just
Okay.
is that it? That's a
lot of
Tina
the times
4.
when we get on the seabed.
Yeah, definitely. I'm pretty dang sure.
Waypoints. We're at the first waypoint,
Oh,
6
wow.
hours, there's some cool stuff.
Another one.
Wow.
Oh no. Oh, I'm sorry.
So cool.
We're just making everyone lose their lunch. Oh. Yeah, we kind of made that one very unhappy. I think we should leave it alone. Let's keep going.
Let's keep going.
Yeah, maybe it will recover if we go away.
Yeah. Just pilot. I got all the other stuff. Go pilot.
Okay. Still in your job.
Siphonophore, I think. Sorry, I have my thing on so I would see. She's just sending photos on her phone of this TV screen with snipe eel. I think I got a better view.
Something
Hmm?
here. There's something
Yeah,
here.
probably. Oh, it's wow.
What is this?
It's like
You can zoom in. Zoom in if you want.
Is it? I think that's its own pigmentation.
This looks better.
I don't think we want that, do we?
Probably not.
Yeah.
Up to
No.
North Point Bridge.
It's not like it's a big biomass dollar.
Yeah.
It's pretty
I don't want to like take things that, you know,
yeah.
are not. critical?
Oh, look how big it is. How big is that?
One looks,
Look at right
it
there.
looks huge. I know, I'm looking at it next to the jelly jars. Is it that big?
Yeah.
Wow, it's
the size
so.
of the.
Like, yeah. It's like the size of a jelly jar.
Wow.
No, it's probably like this around.
Let's get it.
I just think it's not, that one's not really critical for our science, so I'm kind of like, let's
Very
let
cool
it.
though.
Let's let it live.
I mean, that camera on it is just unreal.
It's so beautiful.
Let me come closer.
Oh, I guess you're right. It is like bigger than I thought. I mean, it's huge.
Where are you looking?
Like,
Oh,
would it
wow.
even fit in the jelly jar?
Oh my gosh, it's
Yeah, but
so
barely.
big. Yeah, we couldn't, I don't think we could collect
We
that
should
thing.
leave that all.
It's so beautiful. It's so big. It's huge.
Yeah. Cool.
There I was thinking it was like a centimeter.
I know, I thought it was really small. It's really
It's
big.
huge.
Incredible.
Yeah.
Very close.
Oh my gosh.
Wow.
Very close.
Should send a picture to Steve and see.
I know, someone might.
I know, Steve would know.
Would he appreciate?
Oh yeah.
I don't have Steve's. Can you send it? I can send it to the group. Yeah.
Oh,
Nice flying there. Very fun.
You want me to send it to Steve?
Oh, I was going to.
Beautiful.
Oh, go for it.
Yeah, that was cool. Got like little polypes hanging on there.
Oh yeah, look, there's amphipods on it. Do you see those, all the white things on there? Those are like amphipods that probably just live on it.
Wow.
Pretty rad.
Yeah.
Very cool. It's very
like
cool.
veins. It's almost inside
Updated
the jelly
waypoint bridge.
jar, just to let you know.
What's that?
The jelly jar.
What about
almost
it?
inside it.
Oh no. No, don't do it. I know it's landing on it. We're not,
Wait, where did that come from?
we don't want it. We don't want to
different
collect it.
camera.
See it for behind?
Oh, it's right next to it.
It's too beautiful.
That's a shot.
It would barely fit in there.
The legs wouldn't, but the head would.
Cool. Nice fish. Well done.
Happy.
Yeah.
Happy days.
Mm-hmm.
It's my neighbor. Happy neighbor. Nice one, Leo.
Okay, let's go. That was interesting.
Voice?
Oh, someone's asking how big it was. We thought it was small, but then we could see this other angle of it almost on top of the jelly jars, the collection containers, and it was as wide as one of those. So it's like, how wide are those? That's probably like 8
Size
inches.
of our head, maybe?
Yeah,
6 inches, 8 inches.
yeah, at least this, like the size of a head. I get, yeah, it was big. Ooh, can you stop? Can we see if it's possible to collect that? I'm thinking they're going to be way too fragile, but let's see what happens. Whoa, yeah, it's in. Oh, no, it's not in. My perspective was totally wrong.
Oh wow, look, it's close.
I think it's gonna break apart. But I was like, well, let's try. Let's do a little practice. And I totally broke apart.
Too
Yeah.
fragile.
Yeah, too fragile. Well, maybe we'll get one.
It's good to practice.
Okay, my husband, maybe, I'm not sharing on anything, he says AI says it's a bloody belly conjo.
Oh, it's definitely not.
No, it's definitely
Like
not that.
I'm not an expert, but I do know what that one is.
Ask ChatGPT.
Update.
Yeah, it's definitely not that.
Got it.
Because we collect those for the aquarium
Yeah.
all the time. Like that's a big thing in the deep sea exhibit at the aquarium. And I do know which ones those are. Yeah.
I'm sure on AI.
I
I think I saw something.
feel like blood belly comb jelly is one of the only ones that I know which one that is.
That is a common name, so
It's like the one that we are always collecting with the aquarium.
I think they display it at the aquarium.
By the way, this is very fun for us. We don't get
Yes.
to do this for a whole shift. Usually it's just on the way down or on the way up.
Oh,
'cause
cool.
Everything's at the bottom.
Yeah.
So this is super
I'm-
cool.
I'm glad.
Yeah.
Chasing.
Yeah.
Yeah, I'm not into.
Oh, there's a squid above us.
Oh.
And Ella did say if you can collect a squid,
Go
but
up and see if you
go
can
for
see
it.
A big one.
Where?
Is it? Go up, she said.
Up?
Up.
Oh,
I
no,
don't-
it was like I saw it next to the cable.
Oh, behind.
yeah, okay.
Is that behind
Behind
us?
us.
Oh, is
Behind.
that behind?
Yeah.
Yeah, it's looking backwards.
Oh.
So, yeah.
But that is above, right?
It is above.
Is
It's
that
at the top of that. Yeah, it's right at the top of the foam
Yeah.
block looking
No,
backwards.
there was another big squid up there.
Ah.
Place. There's.
Is that a fish or what is that?
Yeah, let's
Oh,
see.
no.
Probably.
Yeah, that's a
Can we?
Acting like a fish.
It might be a viper fish.
It's going down.
It's
Yeah.
it's
Yeah.
swimming down
Yep.
Yeah.
with its head up.
Uh-huh.
Wow, it went really fast.
Yeah.
Fast. You can't catch up.
Like, nope, I'm out of here.
They're definitely more agile than ROV.
Okay, well, there's a little red guy.
Go for this or keep going?
Maybe keep going.
Keep going.
We're trying to get to 8:30, and then we'll like, yeah. Well, we need to recover the ROV on the deck by 12:30,
So
and
quick
we're
math is from
12:30.
no 2 o'clock. Sorry, 2 o'clock.
from 1,000 meters, we'd have to leave or start heading to recovery at 1,300, and from 500, it's
Yeah.
1,320.
Okay. So we have like an hour-ish
Yeah, a little over, probably an hour.
to search more.
Because we keep coming up as well, so
Yeah, yeah.
time goes down.
So
What's up,
we
Paul?
just keep getting distracted.
Mine used to be 1,400.
Huh?
Ooh.
1,400. That's the one.
They have other things to do today. We're not the focus, is to get used to it.
Very much from this.
Anyone wants to be reinforced?
Yeah, man. Obviously, yeah. I'll explain what's going on. We haven't sampled anything yet. We haven't done Niskins. We're on our way up to about 830 meters now. And then there'll be a couple of Niskins trip, and we're looking for some animals in that area to hopefully collect. The thing things to keep an eye on. You gotta be really on top of where the vessel's at.
The
And
current
delta.
is
Because the current's drifting us this way, so I'm just continually bringing the vessel this way. So, I'll get this set up in the next
Ooh, keep going. Keep going.
Keep going.
Keep going.
Oh, okay. We can look
Okay,
at
wait.
it. Let's look at it. But are you gonna? Yeah.
And then I've been running the camera for him.
We already looked at one of those.
Alright.
We did look at those already.
Got it. I've been keeping the
Well,
delta
I'm
at
just trying to
60.
get us up to the 8:30.
Yeah.
So, and then we can do lots of searching there, and it'll match your water sample, and because otherwise
For around
we're
the
really
south,
probably
yeah.
not gonna get all the
I'll
depths.
keep
So
going.
yeah.
Just skip the Mach Nest toe and do
Yeah,
the
let's
Mach.
just skip the Mach Nest.
The Mach Nest is cool. It brings up cool animals, but you don't get to see them in their native habitat, including.
Yeah,
You don't see them alive.
yeah.
Might have to slow down a little bit if you want to do that at 60.
Because time to time we stop and it's fine.
She says fine.
Carlos, do you see the RV to launch and recover? Do you check that or is this us? Usually we have that distance. Yeah.
RV to
RV to launch and recover.
So there's usually a measurement from where we are to where our launch recovery is. Right now it's
not
50, 60,
showing.
70, maybe like 85, 90 meters, but it's not showing up right there, which it should be.
Is that over? Something
Well, hold on.
I never seen that going away. So.
That's working now, but that still is not. Yeah, I don't know what that would be. So it's not getting. I don't know. Are we happy with that?
I just noticed.
Are we happy with the position?
Yes, we are.
That
USB on
should be fine. We can just visualize it. Whatever it is.
We are coming up to 8:30 meters, but if you see something cool, stop. From there, they're gonna do all the. We didn't do any niskins, any capture, nothing.
Did you guys
Any
change
section.
the Niskins levels?
I didn't.
Alright, but I do it just in case because so you did say 30, but oh yeah, it's yeah, okay, they changed it. That's fine.
The two, let's see. Yeah, that's fine. The current's blowing that way, south. And we are drifting the vessel with it.
Okay.
So, I think that's it. I have the gains low so I can control and stop. descriptive of the weather. It's giving
54
the
is off.
telemetry out of there from time to time.
Okay.
That's it.
Alright, buddy.
Alright, guys.
Enjoy it.
Fun.
One of those episode floors.
Yeah. So we're trying to make our way up to 8:30 and then do most of. Oh, okay. And then we'll do a Niskin bottle and collect some animals there more. More in a targeted way, but if we see.
I have this.
What's up?
I have this. It's just.
That's bad then. Alright, so if there's anything that you want me to talk about, just let me know.
Okay.
If anything like
Yeah,
Alex would want me to look at, that's stuff.
If what?
If there's anything that I think Alex would want me to film,
Okay.
I'll do that. But I'll keep my eye on my delta and I'll keep that where it is, which means we'll get up at the same
Sure.
time. It won't affect the overall
Thanks
time.
to,
Okay.
what's his name? Andy. Andy. Because Andy was saying they're not getting the nav bridge. So they're getting it.
There's a lot of
Talk to
copepods. Lots of copepods.
I don't remember seeing that on the way down.
Yeah. Well, I guess we're moving pretty fast.
Yeah, that's true.
Updated report.
I guess it's probably easier to do when you're doing the calm weather, right? Must get pretty hard when the boat is,
What?
you guys sitting in here.
As far as like sitting right here?
Yeah, just like
Yeah,
if you're sitting here trying to do some really fine work and there's a big wave crashing into the boat,
Yeah, it can definitely jostle us around a little bit.
so we have some nice calm weather for this. Nicer for us.
Updated report.
What's that?
Got any update? Wait.
Or if not, I can find someone else also.
So have you guys seen anything unexpected so far? Or pretty much?
Oh, I don't know what I expected, but we saw. I mean, I don't know if everyone gets excited about this, but we saw a lot of cheetognaths, like so many cheetognaths in the water, which I haven't really seen that before. I mean, you've probably seen that, but I hadn't, so I thought that was cool. We saw a huge ctenophore, like a huge one.
Really?
Yeah, like as big as the D sampler.
Really?
Yeah. It was like reddish.
Oh, was it the they had the two long tentacles on the bottom?
Yes. Yeah.
Okay. Andy mentioned that to me. He's like, or Doug did. He's like,
Yeah.
That sounds really cool.
We sent it to my colleague who is a ctenophore expert, so maybe he'll tell us what the species was. Oh, what's that? Let's see. See that? Oh, is that a fish? Yeah.
Yeah. Oh, cool.
Oof. Can we zoom on it?
Yeah, I'm gonna go closer.
Oh, okay.
Push the lights a little bit. Don't go that way. Don't go that way.
No.
It's really close.
Oh.
He wants to go in the thing. Oh wow.
Oh wow. Let's keep going. We lost it. Up to. We're gonna stop at 8:25. Oh wait, can we look? Sorry. Can we see what? Oh, that's probably a ketognath, right? This big glowing thing. Yeah.
It's a fish.
Oh, it's a fish?
Yeah.
Yeah, it's a little weird fish.
Oh,
We can go look at it.
well. That's a fish?
Were you talking about the one in the front?
This, what's that?
Isn't that a fish?
No, it's a ketognath. There's so many of them.
Yeah. They just keep filling
They're so big. All right, sorry, I got.
Yeah.
Oh, cool. That's okay. We can keep going up. Oh, somebody on the chat knows what the big red ctenophore was.
Oh, wait, look in the distance.
Oh yeah, that's a larvation house, I think.
See, it's worth trying to get.
Do you think it's possible to collect larvation houses?
I don't think that they've come up with any kind of usable form. We can give it a shot.
Well, we just want the organic matter.
We could suction sample it.
Might as well try.
Suction?
Either way.
Isn't a lot of volume goes through the suction? It would get really diluted on the end.
Fair.
As it breaks.
Yeah, I think that probably you have to do it in the D sampler, but let's just try. Let's see. Can we do like a zoom on it or get like a nice image of it before we try to collect it? Oh yeah, look, that's actually, I wonder if there's still a larvation inside of there. Because it has the inner house.
Are you gonna do the job? You gonna do the job?
Yeah, I can do it.
Okay.
If you want.
That's fine.
Fish your poop.
Look at the fish swimming behind
Fish?
you.
not here for the fishing, I'm here for the park.
I know.
Can you see an animal? Is there an animal
To the
inside?
left, it
See,
looks
I think
solid.
it's a freshly discarded one.
Yeah, I don't see anything swimming in there. It's probably like discarded. Let's get it if we can.
That would be cool.
Okay.
Because yeah, it's
Some
pretty big.
excited will come through.
Yeah, how big is it? I don't think of those as being like maybe a centimeter at most, is that?
I think it
That's a lot of
depends
They're
on.
gonna do a test,
Yeah.
but I don't know which side it will come
Just
through.
like that.
I'll go for it.
Yeah, a few inches.
And then there's some like really big ones. I don't know.
Which
if
one are you
they're-
doing?
Okay.
Two?
Alright, go for it. Hold on, symbol. I don't have anything on there. I don't have anything on there.
No. I just want you to collect the hardest things. The most,
Well, before we do
the most impossible.
any of that, that's super slow. Oh my lord.
Yeah, the doors are
Yeah.
really, really slow.
Yeah.
It's gonna be really hard to collect anything. I feel like because they're like
I sent it to Steve. See what it is.
really slow.
Yep, the beds are
Did you see
private.
this night?
Two's not going at all. So two's not working at all.
You see?
Alright.
Yeah, like I mean,
Alright, that's three.
three.
Does the liquid stay in the jelly jars when you bring it on the deck?
Also to
Yeah.
understand this system. It's a cool
Yeah,
component part
I
of
think
the
so.
idea. But yes.
I've never
I
used
never-
it before, so I
They,
can't.
it better, otherwise everything
Two?
falls out.
Different species.
Right, it could be water.
Oh no, yeah, two's worthless. Unless there's something that is very, very still.
So this is a 8 or 902. We're starting to get something in the range of what we see on the acoustics as a high animal density where
Cool.
matches it. I think focused around
I
825,
want
but
to
you
do
know,
a portrait out
confused.
as well. Alright. So we'll do this.
Someone actually has to walk in.
It's a little hot in here. As far as the water going through the jars,
Yeah.
we have that mesh on top. It's pretty
Coarse, right?
Yeah, no, it's not coarse.
Oh, it's really fine.
It's really fine.
Oh,
and
cool.
There are two of them. I put a second one on for support.
Uh-huh.
And when we rotate it, it's pressed up against the plastic on top.
Yeah.
So there really wouldn't be a lot of water going through there at that point.
Okay.
Once it's
Yeah,
going into it, there
right.
would be quite a bit of
But if we stop pumping, then it'll just be sitting there.
yeah. And so yeah, so like, once it was in the jar, I
Yeah.
started pumping, I would rotate it,
Well,
and there would be no path for water to go through.
Yeah, that's like
So-
We could try doing houses that way.
So we can give it a shot. I mean,
But there's been so
I've never actually tried to bring one on staff before. So.
A house?
Yeah.
I mean, it'll just be all destroyed, but if we can like contain the organic matter, then we can sequence the DNA.
yeah, there's the like I was gonna do the biology sample and then Niskins. Yeah. If I would just say I would say like what they did, like our best guess.
I'm gonna turn the cover on and turn the camera on.
We'll have pictures of them.
Yeah, it doesn't matter. Just like jelly.
Yeah. Okay. Yeah.
Hey Lee, can you maybe? Oh yeah, it's got you here. Maybe can you guys just make a judgment call if it seems reasonable to like put electrical tape or something around the bottom of some of those ones that are leaking. I don't know if that's gonna fix it for this, but maybe try. Maybe it won't work. But we, after this deployment, we can try to seal it. We can see if they have any sealant we can use. But for this one, it'll be fine. But if you could try to put something around it, we use them.
Yeah.
We switched out some of them this morning. It's not electrical tape. It's
Well, anyway, is that going to stick better?
I think electrical tape would work better. Because you can kind of stretch it around.
We have rubber tape that we use. Self-amalgamating tape.
What's it called?
Self-amalgamating tape. So,
Where is it leaking?
At the bottom of these collection tubes, of those tubes, there are trap tubes. Some of them is leaking at the bottom.
Like it's cracked?
I think the seal, I think they're brand new, so I don't know why. It's not cracked, but there's a plate on the bottom that's just not perfectly glued to the acrylic tube. So there's water
so it's
dripping
just
at
like
the
a
bottom.
cap?
Yeah, there's a cap on the bottom, and the seal isn't perfect, so there's drips coming out. It's not the end of the world, but we want to solve it at some point.
What if we put like a thin layer of silicone around the...
Yeah, I think we can't do that today because we're deploying them today, but maybe when they come back we could try to do that.
yeah, we can look at, but I would, for one dive, I would try the self-amalgamating tape.
Oh, okay.
Because it makes like a solid rubber seal.
Okay.
With the green. So we can try that. and
Yeah.
see if it works.
Okay. Yeah, that'd be great.
Is it 825?
825 is what Anitra said.
Okay.
And so that's where we'll do a Niskin, and then we can just like drive forward slowly and search for something cool. I'll just tell them that they should get that tape from you.
Okay, so alright. Yeah. It usually goes to this one and senses that one, but yeah. So when we get on deck, we'll
Oh, you're right here.
They're all different things. Some of it is
Okay, so
single Niskin?
2 Niskins. Anitra, do you want to log the Niskins?
Yes.
Okay, just do it.
some of it is 425.
Yeah.
25, this is good.
Well, I mean, there's learning things, but even some of the ground things, if you were to look close, it would be some kind of...
What Niskins were they?
This is D1.
Hey, Sebastian, Chris says we should try using self-amalgamating tape, which they have, to tape around the bottom just for this particular, and then after that we can try some silicone with sealant after we get it back.
If I talk to one of the other ROV guys, they would know that this.
Yes.
What that is.
Okay.
Self, what?
Amalgamating tape.
Okay.
Thank you.
Yep. Alright.
Yeah, but it's 816 now, but I had asked for 825, but this is
1 and 2?
Yep.
bottle of water, you have this one. This is the first one to fire. Yeah, I guess so. We should do 2. We have only got 9.
1.
Oh, yeah.
2.
So did you fired both 1 and 2?
Yes, I did.
Okay. amazing. We never get to tell whether our bottles have actually fired.
Yeah, on this one you can see the top of the flag, and the bottom you can see the strap
Yeah,
down.
it's nice to have that visual confirmation.
Alright.
We had to go.
Yeah. I made them. I made.
Oh, okay. You made it.
They have to prepare our traps because we're deploying them right after
I
this
know, I
ROV
know.
dive.
So 825, just want to go kind of forward to the slow pace. Faster than we were, or around the same?
Sorry.
Do you want to go a little faster than we were, or about the same speed?
I think we just want to drive forward, forward at this depth for a little bit, around 825 or so, and then search here for a little bit to see if there's things we want, because then it would match Anitra's water sample.
Okay.
We're looking for things that that are delicate, that we can't get in the nets, and that we think we could reasonably dissect out their digestive tissue, which some of these, it looks a little challenging on some of these, so. Do you want to look at this one? No.
I mean, I have a good look at it.
Yeah, you already looked at it.
I already looked at
You're
it.
done.
Maybe-
Let's keep
maybe
searching.
Yeah.
Let's keep looking. Because that one, actually, I don't know where we would get the digestive stuff. Yeah.
Speed on Brian, so any damage. What's going on? Oh, that's not gonna work. All right.
So the other thing to keep in mind as we are searching down here is that we need to recover the ROV by 2.
Okay.
So whatever that calculation is, I forgot. It's
Are
like,
we gonna stay at this depth, or are we gonna?
well, if we
continue
have
on?
time to go to other depths, that's what we would like to do.
500 and 375.
Yeah, 500 and 375.
Okay.
We at least want Niskins.
Okay.
We'll try.
Well,
Let's try
why
our
not?
best to collect some animals, but
If we see a lot down here and you just want Niskins up there, we can do that really, really quick. So
Okay.
It's not a big deal.
Okay.
And then, but if this is kind of like, okay.
all right.
So,
Is that Selps? Can we just get a nice video of this?
all right. Can you
I don't think
adjust
we'll
our
collect
lights
them, but
and focus for me, please. Actually, I can do the lights. That's gone. I got it.
we can. If that thing is totally blown away, we can. I'm not sure what direction of the current, like whatever
Current's
direction
pushing-
in the current you
current's
want to
pushing that
go
way.
for doing this is totally fine.
All right. Is it better for me to go into the current?
I don't know. whatever will make it so that we don't like, whatever makes it easier to not blow the
With
thing
the-
away.
Alright, with the current, like it's the side, so I'll just do this. Follow it along to the southeast.
Wow. A lot of cobra pots.
Alright, if we're gonna go forward, we're gonna need to change the distance. So we go with this. So I'm gonna fly forward slowly, but the current's gonna be going to left to right, so that whenever we stop, southeast. So we want we want to go northeast, so that I'm gonna be going northeast. This direction. Okay. Yeah, this time is gonna just change this. Yeah. Hold on. Can I grab that? Yeah, you gotta do that. I can't grab it.
Foundations will like accumulate particles. They
Yeah,
Hey, buddy. Hold on.
they filter, or they basically concentrate
Go ahead. Yeah, you can
small
test the pores.
things through that
You're gonna
house,
change direction.
and
Whenever
then
you head in
eat it, and then they make
northeast,
this
one northeast,
little
we're
fecal
going
pellet.
after
Looks like
you.
a little football, and there's tons of them in the center. There's always tons. Or I mean, or not always, but yeah, they have very distinctive fecal pellets, and it's interesting because they concentrate small things and make these dense aerodynamic fecal pellets that can, basically that carbon maybe wasn't really sinking, and now it's
Right.
And then the other thing is their houses. Once they abandon it, that's just like a big. Those things can sink really fast. Oh, it's solanaceous, maybe. I forgot some kind of a jelly.
It's as far as being able to keep the animal in this direction. So if it's going that way, I can follow it to the right, as opposed to chasing it.
I don't think we want that though, even though it's beautiful, but. Let's are you in a good position with the current? Okay, let's just strive forward slowly. We don't want that jelly.
We had a squid that just flew into our porch a second ago.
Oh yeah, I know it said if we can get a squid, get it. But
They want to say 825. They're like, don't think that. Yeah, it was like it flew right in there. We between like underneath the jelly lids. I was trying to get it clear, but it's not away. It's unfortunate because there are a lot of those little jellies around there.
Okay. I guess, can we see what this is?
Yeah, it's a cluster. It's a squid.
Is that a squid? Oh yeah, it's a squid.
It's a creature. Can I mix with the lens a little?
Yeah, go for it. It's like an owl.
Yeah, look at those eyes. Oh, that looks nice.
Good job, Sos.
Wow.
What'd you say?
It's a good job. I'm pretty zoomed in right now. I'm going to fly a little bit into it.
Okay. I'm gonna take another bottle focus.
Yeah, that's fine.
It's funny how that it's funny how that makes it look like this is the mouth.
Yeah.
Even though I know that's not the mouth, but it looks like it has a funny lips and like a funny hairdo. Oh my gosh. Was Anella joking with me about collecting a squid with the ROV? Because I feel like they would be way too fast,
If
right?
if they don't want to be caught, they would not be.
Yeah.
Sometimes they're just remarkably lethargic. So it's kind of.
Well, I think,
Want to try?
yeah, let's try it. I think it's going to swim away, but I might be secretly hoping it swims away.
Yeah.
But if we can catch it, let's get it.
Are you ready?
Yeah.
I'm gonna go for 3, okay? Yeah.
Oh, it's too slow.
Went through.
It's way
It went-
too
yeah, it
slow.
went- it went slow.
I don't
Yeah.
know about that.
So we have an idea on something. We gotta find it again first. There it is.
If you like half close it or like, I don't know,
Yeah.
that thing is slow.
Yeah. We have an idea on how to mitigate that.
Mm-hmm.
Like what I mentioned, putting a little screen on the bottom, so they could do what we just did.
Oh,
Did
and
that,
yeah,
like,
and they'll just get
do it,
stuck.
yeah.
Yeah. That's true. That would work.
Yeah, beautiful squid.
It's. I can't believe it's not swimming away.
Because we're following as fast as we can move them.
We were moving them the entire time.
Yeah, but then we started going the opposite direction. Yeah, getting behind us.
Trevor, you were point, I'm just gonna bring you closer to us.
Once
No.
it gets in there, it just
Yeah,
vortex.
it's just gonna go straight. It's gonna go through really fast.
yeah. I think what we're gonna have to do is
Yeah.
for stuff like that is what Mike suggested, and that's the Divestream in there that I can do that. It won't go anywhere. It'll be gentle. It'll be a plastic one, so it won't harm the animal.
Yeah.
And then we can close it at that point.
Yeah. Is there anything? Is there some way to make the whatever's closing the door like move faster?
No. So the only way to do that is increase the hydraulic flow through there, and that's not always much as it will go.
Oh wow. Okay.
I'm gonna have to go back over where the boat is, but I'm just gonna look at this real fast.
So what?
I have to go back over where the boat is, and look at this real fast before I do.
Great, can you replay?
Yeah, moving to the one case assembly.
Roger.
Is that different?
It does look different. Can
Yeah,
I
different hair.
take over the camera?
Yeah, go ahead. Hold on, I'm trying to port something.
Oh, there's two little hooks on it, so there's two
Zoom out
tiny-
for me, because I gotta get closer. Yeah, and then try to keep it as steady as
Oh.
possible. Yeah. Alright.
Yeah, just tell me when.
Oh wow.
That's beautiful.
Yeah, it's so beautiful.
I'm not picking I just want to make sure the focus is okay.
Look at those inside. Look at its internal organs. That like you can see everything. Yeah. And those.
Alright, we're gonna have to hurry up and get out of here, guys, because they're really far from the ship.
Okay.
Okay.
You happy out?
Yeah.
We're gonna try to catch
Alright.
that one.
We tried the
Swim out.
desamplers are. It didn't swim out. The desamplers closed really slow. So if we want to catch anything with those, I know.
Oh my
So pretty.
Alright, I gotta go.
Because look
So
how
far
we're
from the
strung
ship.
out from the ship.
Oh, okay.
They gotta get closer to the ship, I think. Yeah.
Yeah.
What you've got on obviously.
Um.
And we're really tight too, so pay me out some, please. And if you could put your dials back on this and see what our distance is. Like 250 meters, 200 plus. Jesus. I'm gonna get yelled at for that. Get my pieces.
What? Given that we're at 800 meters now,
Yeah.
do you think we should? What's your estimate of like if we just came straight up for the ship? Like how long would it take?
40
Like
minutes.
40 minutes? Okay. And then there's another half hour or so for the recovery?
Roughly.
Yeah.
I mean that's like a
Okay.
that's like a longest case scenario.
Yeah, well, let's go with that. Let's do the longest case scenario. So,
Alright, as soon as I get in
a
good
little
position,
over an
can I come back
hour.
up, is that what you're saying? A little bit?
Oh,
You wanna
yeah.
come up a little higher to like 500 or 700?
What do you, Anitra, do you want to keep like looking at animals at this depth, or you want to come up and then search at the shallower depth?
I just feel like we have to go up, don't
So we do, and we're gonna run out. We have
We can.
we have maybe 20 minutes of search
Yeah,
time
so let's
left.
go to 400 and fire a bottle, 2 bottles.
Okay, you want it at 400, not
Yeah.
500?
Yep.
Okay.
Yeah, everything's changing.
Yeah,
I mean, you know.
as
You can-
usual.
You can move his
Exactly.
waypoint like where my hand is right now, and that'll put us.
400 is
that'll give us
the
enough
layer of
space.
densest
Going
animals
further
in the
southeast. You want into it?
400 has
Yes.
the densest animals.
Well, according to the acoustics, it's
Oh,
like
okay.
the brightest.
Yeah. Okay, let's do it.
And
Um,
the biggest animals.
I think even just like the getting some of these close-up shots of things alive and in situ is amazing. And especially, and if they overlap with things that we caught in the net, that's even better, because then we have beautiful images of them when they're like alive.
Yeah, you can actually see their anatomy so much better when they're
Yeah.
not experiencing gravity.
It's so crazy. And at the depth that they're evolved to live at. Yeah.
So I think the ability to get them in the jars isn't going to be a problem. The ability to close the jars while they're still in there is going to be the tricky part.
Looks. I can. I'm not doubting your piloting abilities.
Because if you close the bottom,
Yeah.
then it's probably harder to get them to come into the jar because you have. Like,
Well, what
They can't close independently. But we were thinking we were putting water passable kind of
like a
mesh in
mesh
there,
on the bottom.
but a plastic one so it wouldn't harm the animal at all, and it's small enough that it wouldn't like.
I didn't know right.
I'm just sitting right here basically.
So you want to cross this way here, or you want us to come behind you?
No. He's telling people where it is. That's waypoint's fine.
And then are we also now coming up to 400?
Yeah, we're
okay,
at
great.
794.
Great.
It's unfortunately because once it hit that inside there, it just shot it.
Yeah. I mean, I think that would be a good way to get a few things. Yeah.
Alright, so if we're going to come up, I'm going to just face the naked current. Let's see if it gets brought to me.
Oh, okay. So. For getting so far away.
Yeah.
Okay. I'll be your shield.
I appreciate it.
Science party in the mess was cheering when the animal went into the jump jar.
Well, they didn't see
lunch.
it come out the bottom.
Oh, they did. They saw it swim out anyway. While having lunch, watching.
Oh, that's funny.
It's weird because normally they cheer for the animal.
They cheer for if the animal.
Like if we're trying to collect something that's cute, like no.
Yeah. Yeah. I'm a little bit that way.
They were just amazed. They were just like.
Yeah. Yeah.
Our research that's may just come at the end of this week.
Awesome sauce. Right here. Go.
Pause here? Or
Yeah. You're fine right here. So, yeah, I'm at this heading, so all the currents can come this way. So there's really no reason for me to fly into it as well. I can continue if I see anything, then I can let that little guy. Just have to keep our eyes peeled. That's a lot of current.
All right, someone in the chat cheers for the ones we collect. I agree. I mean, it's important what we're doing, and it's not. We get so much value out of the specimens that we do collect. You know, they're not going to waste. It's like so many people on the ship want to sample them for different things, like animal genomics. There are people looking at genomics of the animals and how they are evolving and species diversity, but then there's a whole bunch of people looking at the microbiomes, like all the bacteria and protists that live on or inside the animal gut, isotopes, metabolomes, the food web. So yeah, they're going to a good cause that will, help us understand all the other ones that we don't collect. That's cool.
Yeah. I'm gonna remember flying off the ship because of the angle. It's another
Oh, someone keeps yawning. I think that's me. I'm sorry. We've had a lot going on. I got like 4 hours of sleep last night, so that was good. It was enough. Sorry, I keep yawning. Someone on the chat is like asking who's yawning. Sorry. I'm fine. I'll make it.
We're almost done.
Well, then we can move on to the deck.
Yes.
And deploy our sediment traps.
Things happen 24 hours a day. on
Oh
these
yeah.
ships.
After those go in, I'm gonna rest.
Do we recover the big black for anything? No.
Yes,
How'd it go?
I think it went well. The mock nest, which collected all these animals. Yeah, it sounds like the mock nest went really well, right?
I think so, yeah.
Yeah. I mean, they're really busy and they're processing samples, so I assume that means it went well. Yeah.
There were a lot of, there was a lot collected.
Ooh, cool. What can we see what that is? Is it a siphonophore?
Yeah.
Yeah.
Very good.
Ooh, ah. Oh no.
Whoa. I have no idea what just happened. From the wind chip particles.
If there's any like Spanish speakers that want to like talk to people, it seems like there are people who would appreciate that on the chat.
Yeah, you never know.
I don't really know anything about the species.
You can introduce yourself and say who you are.
The Chilean scientists. Like, could they
Yeah,
go ahead?
If anyone is free, but they're so busy. But any,
Yeah,
do
you can
any
also
of
introduce
you-
yourself and say what you're
do
doing
you
here.
speak Spanish or Portuguese?
Spanish.
Spanish. Maybe you could just introduce yourself and
Say why
say
you're
what you're
here.
doing here.
Yeah.
Because it seems like there are, like, most of the people on this chat are Spanish speakers. You know,
Yeah.
like, does anyone speak Spanish?
We've had 2 people come in and
Yeah.
talk, but Lucia and Luz were in introducing themselves and answering some of the questions. It's nice for people to know we have an artist on the ship and, you know.
Which microphone do you want to speak in? This one?
Our team, so just so everyone knows, our team is from a lot of different countries. The United States, we have PIs here from Chile. We have scientists from Argentina. Brazil, one of the PIs on the project is from Brazil. I'm sure I'm missing more countries. Who else am I missing? Where's Diana from? She's from.
She's from Colombia.
She's Colombia, but she works in the U.S., but she's from Colombia. But yeah, we have quite a representation. But I'll stop talking so Paula can introduce herself.
Hello, everyone in the chat. My name is Paula Praño Mesias. I'm an artist from Quito, Ecuador, and currently I'm the artist at sea here in the expedition. Hola a todos en el chat. Mi nombre es Paula Praño Mesias. Soy la artista en residencia acá en la expedición.
So if we get to 400 and then fire 2 Niskin bottles. What's that? Okay. By what time should we? Okay, 1:15. It'll only take 15 minutes to get up to the surface.
400
From 400.
No.
To the surface.
Scientist, that's not what Luz is from. Luz is not from Constitution.
No.
Luz? I don't know. She works with Daniela, so I don't know. Actually, I don't know where Daniela works.
En esta ocasión no hay científicos del CONICET a bordo. Pero sí hay científicos que vienen de Argentina.
We're going to go to 400. Get your Niskins and search around, but we need to leave by 1:10.
Yeah, we're happy. I'm just happy to fire the bottles, so that's good.
Everything's running late, so. The main thing is like, that's a good fish. You nice guy. Yeah, we have a course. I bet we'll have a shot at it, but I just want to, it's just the, I really want to deploy my traps. And we need to get that.
Corrección.
Not next in the water by 6 p.m.
A bordo está Daniela A. del Valle.
I don't
Ella es científica del CONICET.
think silly to happen where that mock nest can't happen. Because, and I really want my traps to get in the water today.
Para mi proyecto de residencia voy a trabajar en varios medios, principalmente con performance, con video performance, analizando un poco el brazo robótico del Rob Sebastian. Mi trabajo se centra principalmente en prostéticos como extensiones del cuerpo humano y no humano. Y también me interesa mucho entender cómo la tecnología puede mediar en las relaciones entre especies humanas y no humanas. Así que en esta ocasión estoy investigando cómo el Rob Sebastian puede ser una extensión del cuerpo científico a bordo.
Every one particle in the deep, there's 100 at the surface probably,
Yeah.
in terms of carbon
Yeah.
units. Yeah. So, interesting.
Yeah. All right. You're using something really cool. Sky? What is it? Oh, is it a Tina flash? Oh no, it's a kelp. It's a kelp. That's its, I recognize that fecal pellet anywhere. Mhm.
There's a lot of particles. You'll see what Josh called the last
We're going
Niskin
up.
sample. It was impossible to see what the last Niskin sample was called. Name. What is that something like this?
On the log?
Yeah, like
I
what did
didn't-
he name it? Because he just disappeared. I didn't know what he named the last one.
It should be in the sea log, right?
No, I didn't see one.
Oh. It's Niskin 1 and Niskin 2.
I just didn't see why I
In here it's called, on this sheet, it's NB1 and B2. So it's probably that in sea log, I would guess.
Okay, thank you. Yeah, that's great.
So we're just doing 400 and then coming up, right, Anitra? For Niskins?
I've got also at 200. Two Niskins at 400 and two Niskins at 200.
Okay, really fast. Look at those big blobs. They're beautiful.
So if you want any amount of time, like 400, it's going pretty fast.
What's that?
If you want any time to the ground, like with underground at 400, like come on really fast, just 300 meters to go.
Oh, okay.
So it's gonna kind of zip out a little bit.
We'll just make it quick. We'll just make our looking around quick.
Well, no, I'll go for 500.
Yeah, sure. Okay.
Maximize that.
Okay. Okay.
We were gonna go ahead and let that other thing.
Okay,
And that way we'll be able to surface at 1:30.
That's fine.
And then it's half an hour beyond that.
Okay, no problem, whatever you guys feel.
We
I just wanted
just want to be
know
kind of
what the latest-
conservative.
Yeah, that's fine, I just wanted to tell you what the latest probably was.
Okay.
And I'll be back, I just gotta go talk to them about these hydraulics here.
Tell them that we need more AC.
Yes, definitely more.
You love that kind of thing.
Hmm. Mitra, are we tentatively planning a dive tomorrow too?
He's on the schedule.
Okay. Yeah. So
Daytime as well.
as long as everything goes to plan, we'll probably have a dive tomorrow at the same time. Somebody else.
Mañana, más o menos a la misma hora,
This looks
habrá.
If everything goes to plan, there's a lot of different activities happening, and lots of just figuring out how to make them all work, and also weaving them all in to each other. So, oh, that's beautiful.
Yeah.
Whoa, that.
If I would stop.
Yeah. Oh, we missed it. Then we're hoping, we'll see, we're hoping we can fit in a nighttime for our time zone anyway, ROV dive in the next couple days as well, so that we can see the differences. So we'll be able to see some of those deeper organisms that migrate up at nighttime.
Yeah, I think the one we're supposed to do at night dive at this station also.
Yeah. So that's the goal. What's that guy? Ooh, is that is that an eyeball? Or is that a, no, it's a siphonophore? Siphonophore. I see, it's, huh. Oh yeah, that's the one that we drove past. Because it has those little black or those dark tips.
Yeah, go for it. That's gone.
Oh, it's just it's got
It's still alive. It's just
well,
flowed. Very, very
yeah.
flowed. Very close to us.
Well, and I don't know how they work, but aren't they all? It's a colony, right? So
Yeah.
maybe they just form a new colony if they break apart.
But I saw it down there. It's still like
Yeah. Mm-hmm. Okay. Getting close. What time is it? Yeah. We won't have much time, Anitra,
Yeah, that's
just to search around. We can take it. We can take a quick scan.
Yeah. I mean, I think searching for things in this depth is much easier to find things, but we maybe find really neat things in the very deep water when we're researching.
Yeah, we have tomorrow. Well, in theory, we have tomorrow too.
Yeah. I'm saying we take less time finding animals in this layer than deeper.
We're gonna say.
Oh, I see what you're saying.
Yeah. Location tomorrow.
We're kind of, yeah, I guess so. It's kind of like wherever the ship is, which will still be in this same region.
Okay,
Hopefully,
so
the same water mass.
do we need to do the video transects? No, we can just straight to.
I think so
Search and
because, like, replicates are valuable.
Can you open up the iris phone? ISO, sorry. Two.
Alright, so I'm gonna
Yes, 3 and 4.
this is bottle number 3.
Mm-hmm.
Three more.
And okay, so I did 3 and then we'll do 4.
Yeah. Four. Alright.
Bottle 4.
We have 5 minutes to look.
Okay. Alright.
Yeah,
I
we're
logged-
gonna find
I
the
logged
coolest thing, and
bottle
then we're like,
3 and bottle 4. And so
no.
they're fired and we're good for
Yeah.
water?
We need another 200, correct? Two more?
Yeah, 2 more at 200, but for 400 we're good. Okay. Wow. So much new things. So is that the current?
Yeah,
It's really strong here?
very strong.
Wow. Talk about lateral transport, Kelly.
Yeah.
Oh yeah, exactly. So it's just like the particles are not falling like straight down. They're falling at a very like shallow horizontal.
Yeah, we catch particles in things where they settle into them vertically, but
Yeah,
see
they're not falling vertically.
falling vertically. It's like a blizzard.
Just
And that's what's going to push past us.
Could we maybe get a nice video of that cell?
Is that okay?
Yeah. I think maybe right now, if we see something, if we can just get a nice video, that would be valuable.
Ooh, what's that now?
Is that so? Maybe, no, was that a ctenophore? Or, I'm not sure. That's okay.
I can't keep up with it and not damage it because it's too hard fishing fast.
Yeah, it's fine.
Very choppy, it's a great point.
Go away, moving now.
Mm-hmm. Yeah. Oh my gosh. Wow. Currents are strong. Yeah, this stuff is moving pretty fast. I don't know how we would.
Yeah, it's much harder to get,
Yeah.
close and personal at this depth. Seems like.
I mean, I guess if we were like facing the other direction and drifting with it, maybe.
Yeah. Do we need to
I
come
think-
to 200 now?
I think so. I don't want to
Yeah.
sacrifice. I don't want to get into a Noah's
No,
net time.
I don't either. Yeah. Come up to 200.
Yeah.
Oh,
Of
it's
course,
in
right
there.
when we,
Right there.
right when we were gonna leave.
Oh, wow.
Oh, there he is.
Can you do that direction anymore?
Yeah, and it's swimming. That squid is actually swimming far away from us. He's not gonna just like be asleep and he slam dunked multiple times.
I caught this big once by accident. Well, not accident, but we were doing DNA searching and squids who had a kitchen brush. And we did one and we played the squid buffs, and then
Yeah.
DNA.
Uh-huh.
I just wanted to slash on to the brush, and no matter what I did, it wasn't going anywhere.
Oh my gosh.
And then the science were 30 bucks in it.
Wow. Including the kitchen brush.
But yeah, there's the K-Boss, the kitchen brush of science.
Uh-huh.
Good reference. That's us at all.
Wow. Okay, so the next thing we have to do is fire 2 Niskin bottles at 200, and then that's it for today. I can't believe how many keeling nests are in the water. There's so many.
outside, but the wire is changing all the time. I'm gonna see, I'm gonna gear up and wait outside and just even looking at.
Oh, nice.
Come on, buddy. You doesn't want any part of me?
No. Yeah.
Gels.
Yeah.
We're filling them there because obviously we don't want to put the full volume of formalin brine because the cups are swollen.
Oh.
So just put. Is there
Kinda
approximate
doesn't matter.
volume? And then is there any technique for not disturbing the surface?
You can put 250 is fine. It's totally fine. And
So it's okay to overflow.
oh yeah. So actually, I haven't done this too many times. I would just yeah, so I would do the 250. It's totally fine to overflow, and then just get maybe just put the tube at the top of the jar, not
Yeah.
into the jar.
We got
And-
scared to stab it.
Yeah, I hadn't thought about it. We've only done it like
Okay.
1 or 2 times before. It's not so. And
Okay.
that,
There's no special technique that we need to know
no.
about.
Well, I would say just try really hard to like be slow when you release it into the bottom of the tube. Yeah.
Cool. Well, we just have to do that when we're done. So just
Oh,
those 5.
awesome. How long does that take? Okay, about an hour. That's good.
RV bridge, that's me just coming to stop on the movement.
Roger.
Has it begun?
I was gonna step at 200 into 2 more Niskins, and then.
Okay.
Cool. How do you want to do it?
Where you on where? You want somebody outside the show?
Mike is out there right now watching the miracle.
Is he? I didn't see him.
He's gonna be sitting underneath the.
Okay.
He's gonna gear up and go out there. So
Oh, okay. You want to go out there with him and help him with the belly pack?
Yeah, we just lost the
Jar? Oh, did you have it over there?
No, I had this head up there. I was watching everything.
Oh,
and
okay.
go for majority of it.
Okay.
Yeah, I'll go out there and give him
All right.
some check for
And then he can do it himself.
Yeah. All right. So yeah, 200. Yeah, right now.
200 trip to Niskin?
Yeah, right now I'm
Okay.
up all the way, left all the way to keep this right there.
All right.
So,
Cool. You're doing what? All the way to Depot?
Lateral, short, and up. It's good stuff. I don't have a whole lot of phone.
We just need to fix that.
We just need to stop at 200.
Alrighty. So can you do me a favor? Where the winch picture is right now, can you put the aft camera, which is actually SD camera? It's not an HD, it's an SD camera. That may be it. Yep, thank you.
Sorry, I was operating off of muscle memory.
All good.
If you don't need me here, if you can just make sure those Niskins,
Yeah, I'm logging that.
then I might try to eat something before the track deployment,
Okay.
if that's okay.
That's a
You're
good
gonna have
idea.
photosynthesis when you're out there.
Otherwise, I'm just gonna eat candy like I did yesterday.
Well, yeah, considering you've only slept a few
Not
hours
sound
and you
nutritious.
lunch, you skipped lunch, you should probably go and do that.
Is that okay?
Yes.
I don't think I'm needed at this station
Juncture.
anymore,
Okay,
right?
so are we at 200 now?
Just about.
It's pretty good. That is discipline, it's not less as well, it's not enough. Thanks, Colleen.
You should have those locations and see what's
I did last night, but I could send the most up-to-date ones.
very connected tracks.
It's just going on the same line.
Okay, we're at 200.
Okay.
Ready?
I don't know.
Tripping?
Bottle number 5, right?
Yeah. Five.
Did 5 go?
Yep, and 6. That's all.
Our bottle number 5.
Yeah.
Yes. I can do the sample logging. It's amazing. They don't trust me to do much with it. Six. Okay, so I logged both bottles.
Alrighty.
So yeah, I don't think I have any other time to do anything else, but we need to get to the surface.
That is where we're headed.
Vamos a subir. Muchas gracias por acompañarnos en el primer streaming de esta expedición. Si todo sigue de acuerdo al plan, mañana también tenemos streaming.
Loud and clear.
Alright. Get Mike on the belly pack, near on the deck.
Let us know when you're ready to take control.
He's getting himself together right now.
Yeah. Bridge, RLV.
Go ahead.
Yeah, Ben, I'm gonna probably need you to drift aft with the current while we do this recovery.
Oh, copy. Do you want me to give the deck a shout?
Yes, if you could give the deck a shout as well, please. Thank you.
Copy.
Thank you.
Right now, the umbilical's out 30 degrees to start.
Yeah.
Can you hear me, Chris? Gotcha. Can you hear me, dog?
Loud and clear.
Cool.
Out haulers are selected.
Copy that.
And hello, everybody. That's us finished with today's dive, dive 895. As you can hear, the pilots are talking with the deck team,
You're good.
and we are ready to recover. So that means we're gonna mute the audio for the rest of the ascent, but we'll keep the video on for everybody. And I hope, on behalf of the whole team, I hope that you tune in for tomorrow's dive. And thank you so much for watching today. Keep those notifications on for our channel for Schmidt Ocean, and follow us along for the rest of the expedition. Take care, everybody, and see you soon.